Good evening, dear fellows of Indian Society of Genetics, Biotechnology, Research and Development. I extend a warm welcome to the first day of this fifth national conference by Indian Society of Genetics, Biotechnology, Research and Development in collaboration with Departments of Botany and Biotechnology, St. John's College, Agra. This fifth national conference is on innovations in plant sciences, genetics, technology, and sustainable future, bridging the modern biosciences techniques with AI and machine learning with plant sciences. We have with us our very special guest and the president of Indian Society of Genetics, Biotechnology, Research and Development, Professor Dr. R.C. Agrawal sir, who is the former DDG, Deputy Director General of Agriculture Education, ICAR, and National Director, NAHEP, Government of India. We warmly welcome you, sir.
Thank you. Thank you
Next,
very much.
we have the General Secretary of Indian Society of Genetics, Biotechnology, Research and Development, Professor Dr. P.K. Upadhyay ji, who is also the Conference Director of this fifth national conference. We have with us Professor Dr. Samuel G. Singh ji, who is the Professor and Head, Department of Botany and Biotechnology, St. John's College, Agra, and is the Joint Conference Director of this fifth national conference. We have with us Dr. Niranjan Kumar, who is GIS specialist from Kanha Shanti Vanam, which is the global headquarters of Heartfulness Institute, Hyderabad. We warmly welcome Dr. Niranjan, Dr. Samuel G. Singh, Dr. P.K. Upadhyay, and Dr. R.C. Agrawal, sir. It is a great pleasure that we all have assembled here on the first day of this fifth national conference. And now Dr. Rajendra Kumar sir has also joined us. Dr. Rajendra Kumar sir is former is former DG of Uttar Pradesh Council of Agriculture Research and currently serving as Principal Scientist, Division of Genetics, ICAR IARI. Warm welcome, sir. I would like to invite Dr. Niranjan Kumar so that we can go with our first presentation of day first of this national conference. Dr. Niranjan Kumar will be speaking on geotagging mega project of Kana Shanti Vanam 2025 in collaboration with Horticulture Nursery, Forest by Heartfulness and Heartfulness Institute. Dr. Niranjan.
Hi, good afternoon, everyone. I am Niranjan, as introduced. So we are located in Hyderabad, Kanaha Shantivanam, one of the beautiful place for visiting, the like for meditating. Also, we are doing a lot of research here on the vegetation part. So this is our world headquarters for Heartfulness Institute, and it spans around 1,400 acres. It's very on the outskirts of Hyderabad. So we have around more than like around 1 million trees planted. Like since before inception, it was like a barren land completely. So with our guide, uh, Kamlesh D. Patel ji, so we are able to develop these many trees and with the advanced technology. So we have adopted like after planting the tree, so we are in the thought of like following the growth and where exactly it is located to further develop that inventory, we are using geotagging. So, geotagging in the sense like we will locate the tree exact location with a 2 centi 3 centi accuracy. So till now we have done almost like a 1 lakh 10,000 trees in and around Kanha. This is one of the project near the Kanha. So where we have around 1,500 uh, biodiversity as well as fruit trees. You can see this legend here and the number of trees. So these are all like exactly located in that. So this is again Kanha. Our inside Kanha Santivanam, we have done like entire. We are doing from one side. It's still ongoing project. I just want to show like what are the some. If suppose somebody interested in certain species, we can separate them after geotagging. So here I separated white sandal as well as red sandal. So white sandal is blue color and the red sandal the orange color dots here. And the other biodiversity trees. So around 84,000 or more like biodiversity trees here. So this is again inside Kanha, like there is agricultural farm is there. So we just separated these apple trees, pomegranate, showing the inventory for the persons who are managing this. So this helps them for maintenance and monitoring costs. So this is one more mango garden. Mostly the mango is dominant here and mixed with other trees also, amla and guava. This is a pond near our apartment. So with the initial like involvement of employee involvement program, we have planted around 3,000 plants here. So, okay, these are the works we have done. If you go back to the technicality of the how we do, the hardware we use generally the GNSS receivers. So they like two GNSS receivers we need for this system to work. One will be base station. So and the other will be rover. Base station we place at one place and we'll fix the coordinates like we'll connect to the satellites so that it will fix the location and then we'll connect the rover to the base station. So with the same, if you see the technical configuration, both are same. But only thing is base will be located at one place, rover will be moving wherever you need to take. So and the rover also connect the same way to the satellites. But the why we need to connect these two because there will be atmospheric disturbances. So this will nullify the atmospheric those disturbances and will give the exact location. So the software generally we are using Emlet brand here, the recent model RS3 and the same rover also. And we are using the software. They want software like a cloud soft, cloud software which will support our cloud database also Emlet Flow. And GIS software we are using ArcGIS, ESRI ArcGIS. And for process, for processing also Emlet Studio we are using. This is the workflow, how it goes from like a project preparation, base station setup, field collection. Once field collection is done, like we will go and collect the location of each plant, its name and scientific name, all this. If needed, like sometimes if something like agricultural project is required, so we will take the height of the plant also. So then we'll move to the data management and integrate with the GIS. So this is one of the like a photo source like we are doing the geotagging at one of the place. Like this is the base, yellow color you can see in the front. We have set the base here. It will not move, and the other two guys are carrying the rover in the back. So they are taking the geotag of that white cotton silk. And there are two options. If you go to like a technicality, like it works with the radio that is LoRa mode. And suppose you want to do some distance, like a long distance plans also, then you can use. It's always better to use the SIM card that works under N-Trip. So these are all like inside that operating system some settings. So N-Trip will work up to 40 kilometers. LoRa will be little less, like around 10. Like we tested up to 7 kilometers. It was working. Yeah, same thing. Tree data collection. We will navigate to the plant and it will collect XYZ, like latitude, longitude, and the tree ID we will enter manually. So then we will move the data like once collected, we will import the data like in the CSV, KML, save file. It has the provision to export the data into these many formats. Once the data is exported, we will import into GIS format. So wherein we can do a lot of what you call analysis, like a plant distribution, how the like plant is wise, like a different data we can allocate to that. Here we can see the uh, how the database looks after importing into the GIS. So first one is the common name, next scientific name, the easting, northing. Easting, northing will be generally this is the UTM coordinate system and elevation. And if you want something like geographic coordinates, generally they use geographic for Google mapping and all. So they use we are giving longitude and latitude also there. And the accuracy we have measured the accuracy also like a back check. There is a option for stakeout. Sometimes it will show in the what do you say that in the display in their app itself it will show how much accuracy it is. So mostly we are getting 1 to 3 centimeter horizontal and vertical 2 to 5 centimeter. And sometimes the plant is inside, so we can use the tilt compensation. So we don't need to go there. So we can put the end of the pole. So the tilt compensation, if it is below 60 degrees, it works fine. If it is more than 60 degrees, the error will be increasing. So these are our guys who are doing the geotagging locally or in institutional land. So that is the manual geotagging process. So there is another thing we are applying. Suppose if we know like a in agroforestry region, entire region is one single species, then we can use the drone also. So with the drone, we can like if we fly the drone, set up the mission where it has to fly, and then it will go to the location. It will fly. But before that, we have to connect to get the accuracy, location accuracy. We have same thing. We have to connect to the base station before we are connecting the rover, right? So here drone will work as a rover. Instead of manually person carrying the rover, drone will be working as a rover. So it will go and collect the points. So here we are setting up the drone, connecting the drone to the base station and then launch it. The moment you launch, it will go line by line. You can see how the photos, these red dots are each photo. Like for the entire Kanha, we have around 4,600 photos at a height of around 100 meters. So like it is giving resolution up to 3 centi resolution. So when you process like the second black background window is we are processing using photogrammetry software. So which will a kind of stitching like all these photos, it will stitch together to form a single big image. So even if you zoom, like here we cannot accommodate, right? So if you zoom it, it will show like a 3 centimeter, like almost like you can see small pebbles also. So much detailed information we will get it here. So benefits of using these drones, it's like a 5 times faster compared to general conventional service. Who requires less manpower, can access the unreachable areas, suppose the hilly areas and sometimes like deep forest areas, it's difficult to access, right? So we can do it. Of course, we will not get the small trees below the heavy canopy. So no need to close the highways. Sometimes for surveys they will be closing, right? So here we can do like this is generally for the road surveys, not for the geotag. Can generate multiple products like at the with a single drone flight we can generate orthomosaic image in the previous slide. So the last one is orthomosaic image here. So and the point cloud which will give the heights also, 3D model, the digital terrain model, digital surface model, digital elevation model. You will get the contours. And also, if you have the sensor, based on the sensor, suppose if you use multispectral camera in the drone, you will get the NDVI also, which will like with the NDVI, we all know like we will be able to see the stress levels of the trees also. So this will help us for the effective planning, like sometimes.
Hello.
So by observing all the details from the drone itself, like using where all the plant is stressful, so we can back check why it is stressful, and we can alert the person who is monitoring, the local supervisors there. So key enablers for effective planning are dam and contour lines, which will give the slope, so you can plan the irrigation system accordingly. So, and the orthomosaic image provides high resolution, detailed information of the entire terrain, including landscape, gaps, existing plantation, infrastructure, access roads. Like all the inventory we can do, like instead of going to the ground, we can do desktop survey. But because so detailed image is already available, and the DSM DTM will help to check the plant. So DSM is like a top surface which is visible to the drone, and the DTM will average based on the difference. So that will DTM is the bottom actual terrain model. So if you separate DSM minus DTM, you will get the exact height. Like there will be little error, so we will get more or less like a five, like a 20 centimeter difference. So the plant heights we can extract here. And the NDY, as we already discussed, we can use for the like stress to find the stress levels in the plants. And this is the one of the image which we done in 2020 for the entire Kanha campus. So if you see this here, this is the institutional land beside our Kanha main campus. So the left one is the actual orthomosaic image. Second one is the NDVA. Third one is the DTM terrain model, and the last contour levels. You think I was fast?
Thank you very much, Dr. Niranjan sir. It was wonderful to know about this GIS support that is happening with Forest by Heartfulness in Kana Shanti Vanam. I would be requesting Professor Dr. R.C. Agrawal sir to please comment on this presentation and give his views on the same.
Thank you very much, Dr. Anshman. So first of all, I want to compliment ISGBRD for organizing this conference on a very important topic, and that includes the latest topic of innovation in plant sciences. So, what is the use of AI and other kind of things? So, thank you, Dr. Padhya, to you, Dr. Anshman, to you, and the entire team, St. John's College. We see the head of the department there, Niranjan. Dr. Niranjan, this beautiful presentation, I think we should compliment you, Daji. We should compliment the entire team at Kana Santivanam. And this particular kind of thing is really required at the initial level. If we can really geotag each and every plant, so it is not only the cost effective, but we can take care of our total vegetation which is required. The deforestation, we can know that how much deforestation already we have done. Because, in today's context, the resources are really depleting. The natural resources I'm talking about. Uh, if you see the land availability, I think about 20 years back it was around 7 acre per person available at the global level. But today it has come to 4.3 acres per person. You can see how much load we have got on this Mother Earth. So the natural resources also are depleting. Everything is really going down. So we have to take care of this technology. How really we can map everything, like the one you have told through NDVI, through other maps. We can know exactly and then take the decisions that how really we can go for more and more kind of vegetation. The intervention for the benefit of my other colleagues, Dr. Rajendra Kumar, uh, and my other colleagues, I can just say that the Kana Santivanam initiatives for this vegetation, initiatives for this forestation, not only at Kana Santivanam but at other places also. I think Kana Santivanam has got a lot of projects in Madhya Pradesh. They are doing some good work in Gujarat. In Delhi also, there are some talks going on for this complete RIS for again beautifying, doing forestation. And I know a couple of other places where this great work has been done. And this entire work is being done by persons who have been really at the helm of affairs when they were in the job, like Dr. Ramakant, or the one you see, Dr. Niranjan, who has just now presented. And you see a lot of other persons, Dr. Sai Kumar, who have really put all efforts to make that as a model. So that model really we need to capture and really want to see. So my compliments to you for this great presentation, great work which is being done at Kana Santivanam. And next time when I come to Kana Santivanam, I would personally interact with you and just see this geotagging work. Very fascinating, and it's a
You're welcome, sir.
very
You're most
important.
welcome, sir.
Yes, most important. Now about this conference, I can see the great work again by the team. That how really they are connecting different persons on this innovation in the plant science. In today's context, we see the importance of this artificial intelligence. There are positive aspects, there are negative aspects, but always anything you see, any innovation comes with all these two aspects, positive and negative. But we have to make a balance between these positive and negatives. Presently, like I can say, if you go back around 30 years back, when I was doing my postgraduate program in 1983, and we were taught computers. So that time also people just thought that if the computers comes, then there will be a lot of unemployment, and people will be out of job. And if you go for the railway reservation, then all those clerks they will be out of job, and will be very difficult. But people did not realize the positive aspects. Now, if you just say that we want to take out, we want to again go back to the manual system, and we want to just revert back, so people will start opposing now. So the same thing is going to happen with the AI in future. We will all depend upon this artificial intelligence, and will be greatly dependent upon this. And I always tell to the students, young students, that whatever is your discipline, please get ready for the education about this AI. So be in touch with this AI. Just see what are the innovations happening, how you can use the AI in your own field, whatever is, maybe plant pathology, maybe genetics, maybe economics, maybe it is your soil chemistry or physics, whatever discipline you have got. But in plant science, you must see the use of this artificial intelligence, uh, because I always believe that the mother of opportunity is the innovation. If you do not capture this opportunity at the right time, time factor is also important, then I think you are left behind. So if you want to see really the innovations, I think again I can say that you must go to Kanha Santivaram, can see a lot of innovations there, and especially in the plant science, in the agriculture field. How you they can do the mosquito management. And yesterday I was just seeing a news clipping that in Brazil there is a farming of mosquitoes. So they are breeding the mosquitoes with a particular bacteria which they are injecting into this, which controls the other dengue and this other disease spreading mosquitoes. So they are controlling mosquitoes through mosquitoes. So that is again innovation. If I talk for the innovation, I was just reading one article that this drip irrigation, how it was invented. In Brazil, somebody saw that when there is a scarcity of water in Israel, and they could see that few plants, they were very green and they were just growing very well. Then the person observed that there is a leakage in the water pipeline, and then he came out with the idea that there can be something like drip irrigation. So it is how you observe the things. It's a opportunity. So those kind of opportunities our students really have to see. And if you see one of the report of this Niti Aayog, it says that by 2030, so that means just after 5 years, we will be able to feed only 59% of the population if you do the farming the way we are doing. So that means if you do not use the technology, if you do not use this drone, if you do not use this latest technologies, innovations, the latest seed replacement rates, if you do not use this fortified varieties, many other kind of things which have been evolved, you will not be able to feed. So you have to use this technology, and that is why this conference is focusing that how we can take help of this innovation in the plant science, and using that only we can really feed the emerging population. The only thing which is the greatest threat to this earth is this natural resources. uh, कल मैं एक I was just reading one book on Birsa Munda. I think you all know Birsa Munda. There is a agricultural university also in Jharkhand, in Ranchi, which is BAU, Birsa Agricultural University. So I was curious to understand what is who was this Birsa. And you see the kind of innovations he made, the kind of contribution he made for that particular area. So that is really remarkable. He died at the age of 25 years. And by the age of 18 years, he was knowing about all the herbs which can be used for the cure of the human beings, not the allopathic, but through this Ayurvedic. And that's why he was regarded as a god. And he's in Jharkhand. If you go, you will just find the boards, Bhagwan in front of his name, Bhagwan Birsa Munda. So that kind of knowledge, that kind of really wealth, that kind of really this uh, natural resources we have got, but we are not aware. If we go for this technology, if we really go for this innovations, the use of this GPS, use of this geotagging, other kind of things, and it's not only that we can know about our strength, we can further do the research on those species. We can really go for more and more use of those things. Still, I am of the firm belief that this Sanjeevani Buddhi, which has the reference in our Ramayana, the Sanjeevani Buddhi is still available. A lot of Sanjeevani Buddhi is available on our Himalayas. The only thing is we are not much aware about this. It is full of God has given a lot of natural resources to Indians, and we are still golden bird, सोने की चिड़िया जो हमें कहते थे, आज भी हम सोने की चिड़िया हैं, because in nowhere other than India, you can find all kind of soils which are available to us. You cannot find all kind of irrigation systems like so many rivers you don't find in any other country. You don't find so many climates which are available in India from plus 52 to minus 52. You can find. So we can be really Vishwaguru. We can be really the leader in every kind of activity. The only thing is we have to know our strength, and to know our strength, uh, we can use really these technologies, and these are wonderful technologies which we must really take care. We must implement, and I will once again compliment the society. I will once again compliment the speaker uh, and the other colleagues especially my colleague Dr. Zain Kumar for being here and to me to give this opportunity. I'm really thankful to you. Thank you very much. Anshuman.
Thank you very much, sir. It was great listening to you. And thank you very much, Dr. Niranjan, for joining in and letting all of us know about GI taggings, about drone technology also. Uh, I missed if we to tell everybody that Dr. Niranjan is a specialist in drones as well. So if you want to learn anything on drones, you can reach out to him directly. And I personally suggest, because I have visited Kana Shantivanam several times, it's a wonderful place to learn on about agriculture, about botany, about plant sciences, about forestry, and a lot more. It's a place where you'll find everything under a single roof. And without wasting much of the time, I would like to invite our next guest speaker, Honorable Professor Dr. Rajendra Kumar Sir. Rajendra Kumar Sir is former Director General of Uttar Pradesh Council of Agriculture Research, Lucknow, and currently serving as Principal Scientist, Division of Genetics, ICAR-IARI. Professor Rajendra Kumar holds postdoctoral degree in agricultural biotechnology with specialization in genetics, plant breeding, and molecular breeding. Professor Kumar has multiple organizational experience in various capacities in SAUs, ICR, both systems, and several international organizations. He has made outstanding contribution in the field of agricultural research and education by way of commercial release of 20 varieties of pulses and vegetables, development of 50 technologies, publication of more than 1,000 journals of international repute, in addition to more than 200 other publications, operation of 32 research students for their thesis work, and organization of more than 33 international national congresses, seminars, workshops. His overall API points UGCCS format has come around 5840 with Google score citation of 1569, 1569 H index 20 and I10 index 50. Professor Rajendra Kumar has demonstrated his academic leadership capabilities by establishing ultra modern research facilities like Phytotron, genetic engineering labs, and initiation of diverse research. Academic endeavors, including PhD program in biotechnology. He has expanded activities of UP Council of Agriculture Research, that is UPCAR, by staff strengthening, budget mechanism in place, implementation of 5 years plan for agricultural research and education, publication of quarterly UPCAR newsletter, and conduction of hybrid seed trials for private seed companies, in addition to conceptualization, invitation, getting design, and finally funding of numerous research areas projects. He is also the founding president of the Uttar Pradesh Academy of Agricultural Science. Professor Kumar has provided a vibrant agricultural think tank for the Uttar Pradesh state and has been conferred with numerous prestigious academic recognitions by Indian Science Congress, INSA, NASI, UP Government, DST National Award for Promotion of Science and Technology 2019 on the occasion of National Science Day celebration on February 28, 2020 by His Excellency President of India, and the latest one by ICR AICIP on Ravi Pulses Best Center Award 2024. On addition to this, I would like to bring into the notice of everybody who has joined us that Professor Rajendra Kumar sir was also the chief guest for the second international conference of Indian Society of Genetics, Biotechnology, Research and Development, which was held in 2016 February at St. John's College, Agra. So we are again with the same division of St. John's College here. And now I hand over the mic to Professor Dr. Rajendra Kumar sir.
Thank you, Dr. Upadhyaya, for very lengthy introduction. And before moving forward, chair of today's event, Professor Agarwal, St. John College, which I visited long back in 2003, when I received Dr. Salgare National Award there in the campus itself, uh, with the hand of then DG Indian Council of Agriculture Research, Dr. Bangla Rai. And of course, I have association, very close association with the Indian Society of Genetics, Biotechnology Research and Development. So now I come directly to our interaction. So of course, the area chosen by Dr. Upadhyaya, as already explained by the chief guest of the event, Professor Agrawal, that is very timely and actually need of the not only nation, rather global requirement. There is an urgent need that there should be some innovation, especially in the plant sciences. Plant sciences had its own importance in comparison to the animal sciences. Of course, at the global level, animal consumption is not less than plants produce, but even then, in many aspects, plant produce are always supposed to be more beneficial as well as economic in comparison to the animal produce. Now, as we know, in the Indian scenario, pulses are one of the a very important component of our daily diet. Of course, in the ICR system, each and every scientist is supposed to work only with a crop. So right now, I am working with the chickpea, that too the focused one. Right now, I'm working on the chickpea nitrogen fixation. So I thought that I should not take much pain in preparing the deliberation. So I remain prefer to confine to my own activities. So accordingly, when I joined back to IRI in 2018, I wrote several projects and I was lucky enough to have one DST SERP sponsored project that was association mapping for nitrogen fixation utilizing global chickpea germ plants. So out of that, we have derived a very good output. With all these in background, I'm moving ahead. Chickpea, as we all know, is very vital and essential legume crop, and it has its own agricultural and economic importance with a relatively large genome size of around 738 megabases. Chickpea roots establish symbiotic relationship with soil microorganisms. A number of microorganisms are always available in the soil layers, as we human beings have more than 3 trillions of microbiome in our own gut system. And out of those microorganisms, if we can integrate some of the genes which are very specific as far as nitrogen fixation is concerned, and they can be manipulated utilizing the genome editing tools. Certainly, there is more scope for enhancement of the yield potential of not only chickpea but pulses as a whole. The reason behind enhancing the productivity is, as Professor Agrawal mentioned, that our resources are crunching with every passing day, rather every passing second. At a time, rather not at a time, at many fold, I always used to say that our resources are sinking, and even in presence of the QRT team, I spoke before Dr. Mangla Rai that now IRI needs to be very focused. Rather, it should focus for the vertical growth rather than horizontal, because almost all national and international organizations are going on the cost of IRI. So I will not divert the deliverance to IRI, but what I mean to say, there is a need that we must enhance our productivity almost in the all crops. So here, our concern is how to enhance the productivity, overall productivity of chickpea. That's why I'm talking about the symbiotic relationship. As far as global temperature, global calamities are climatic threats are concerned. We can see that there are several reports that global temperature is rising every day, and by 2100 it will rise around 5 degrees centigrade. Accordingly, with the increase in the global temperature, global hunger will also rise to around 840 million of the world population. Very recently, last year in California, there was a wildfire. As we all know, it was the buzz terminology in almost all newspapers globally. Very recently, last year only, we could see there was a district village in the Ganderbal of the Kashmir. Accordingly, the already Khelo India Winter Games were postponed in the very recent in that was scheduled in February 2025, and now we are observing the heavy rain caused flood devastation in North India, spreading from Himachal Pradesh to Punjab, Haryana, Uttar Pradesh, and Madhya Pradesh. So now, in light of these climatic threats, either in the form of the rising temperature or very low erratic temperature, as well as heavy rainfalls, so and so, several forms. There is a challenge before the scientific community, not only for everything. Even Dr. Agarwal mentioned about Brazil has initiated some research program to breed for the mosquitoes, and those mosquitoes can be utilized to kill certain types of mosquitoes in order to curtail the spread of the dengue, etc. So, but the challenge before us is how to feed the 10 billion worldwide population by 2050. That's a challenge before us. And this challenge, nobody else will meet, but only plant scientists have to work hard, have to think in different way, and we have to tap the areas which are still unexploited or untapped. In order to that, especially our team under my leadership, we have developed 20 release varieties, and out of those, 7 varieties are chickpea drought tolerant, and number of the good varieties are there, but only these varieties are not going to resolve the issue which are going to face. So that's why this is one of the area in case of chickpea, because we all know that chickpea is a crop which is capable to fix atmospheric nitrogen ranging from 60 to 140 or 43 metric ton RKG. So in out order to that, I thought that this area is unexploited as far as chickpea is concerned. So I had visualized and imagined to write a project, and this project was very much debated because in the review committee, microbiologist said, no, no, this is the area of the microbiologist, how a plant breeder can work. But anyway, plant breeders, I was not there, but reviewers educated vice versa, because the project was very much in the peri of the crop improvement programs only. So I was lucky enough that I could win that project. So later on, different studies we have already worked in as far as chickpea is concerned. I am presenting my work only confined to the chickpea here. Genomic literature we have published in 2013. Genetic resources for the dope genes we could be able to publish in 2016. Drought responsive genetic and genomic resources we have published a number of papers already in the public domain. Accordingly, we could also publish a good paper in the area of nitrate reductase, which is supposed to be one of the antioxidant. Accordingly, super oxide dismutase, once again very good antioxidant. These two, nitrate reductase and superoxide dismutase, can contribute for the drought resistant varieties in chickpea. And very recently, last year, 2022, we published one paper on cytokinin dehydrogenase. Once again, this is one of the very good gene which can contribute for the drought resistant develop chickpea variety. And we have also compiled or given our hypotheses what strategies can be in the public domain and how we can modify. We published two papers in. I'm talking about these all high impact journal papers. And very recently, we have published five papers in case of chickpea symbiotic nitrogen fixation. One by under my leadership by Chandra et al. 2023, and two papers more in 2024. And accordingly, in 2025, we have published two papers on the in the area, and our two papers are in pipeline. So I think that will be the end of my academic life, because in very near future my retirement is due, and probably I don't know whether in the societal mode or any other opportunity I will get to have any project. Anyway, that's not my concern. So finally, the food exhaustion needs and amalgamation. This is one of the potential area which I see. Actually, conventional plant breeding and novel methodologies have to be integrated, particularly gene editing. Unless until these latest technologies are integrated with the conventional plant breeding, probably we are not going to resolve the issue which scientific community is going to face in the future. So on this aspect also, we have published two papers. Of course, these are the hypothetical papers, because still I don't have any facility to work with the gene editing, but theoretically and hypothetically, I have envisaged that this technology can be integrated, and these are the need of the hour also. So now, very recently, I have published one paper in the Frontiers in Plant Science that was published in 1st September only. We had taken a set of 20 genotypes that we extracted from the association panel. That was extracted from the core germplasm, core germplasm that was global core germplasm that was constituted by the Dr. Upadhyay et al. At ICSE. And these 20 genotypes were subjected to uh, NPK, BAM, and uh, other uh, rhizobium cultures. And these different combinations of these nutrients were mixed, different combinations are prepared. That way, we could be able to have 8 treatments. And these experiments were conducted in a randomized blind design. So we did experiment in the parts as well as in parts in the normal condition, normal climatic condition, as well as in phenomic compounds also. As we are lucky enough that IRI is having a very good phenomic compound. So in the process of that, pre-planning soil preparation included application of fertilizers and biofertilizers, so and so. I'm not going in detail. So, but we had a large our intensive studies like descriptive statistics, correlation, path analysis, CRISPR component analysis, agglomerative hierarchical clustering, and gene expression studies. These were performed. Among treatments, NPK and rhizobium combination significantly enhance the nodulation across genotypes, while the rhizobium plus BAM, which nomenclature at treatment 7, identified one genotype ICC 9085 as a superior donor for the number of nodules, aiming for the sustainable chickpea productivity. And finally, what we did, we also performed the gene expression profiling utilizing QRT-PCR, and we could decipher that rhizobium plus BAM treatment notably upregulated several key genes, including CANFP, GST, uh, leg hemoglobin, nodulin 6, and TAL-YK3, with CANFP emerging as a so out of these five genes, CANFP emerged as a pivotal regulator of the nodulation because it was having the up regulation, and this can be one of the potential target for enhancing some symbiotic nitrogen fixation efficiency in case of chickpea. In the phenomics compound, of course, we utilize the phenomic measuring instrument, and we compare two of the genotypes which were having the maximized nodulation, and other was the lowest one. The lowest one genotype was ICC 83, yeah, 9083, and the highest one was having 9085, as I discussed earlier. So we we for the both of genotypes, we moved at several steps, and we can compare. This is for the 9085. Here we can different phenomics just for the visuality. Otherwise, I have summarized all this data in the last in the tabulated form, uh, table form. So now this is once again this is 9083. We can see in the same stage for the treatment 1 only. Accordingly, the treatment 2 for both of the genotypes we have compared. Now for the treatment 3, once again the 9085 and 9083. So here we can see difference. Accordingly, treatment 4, once again, and accordingly treatment 4. So vice versa, we have, I am presenting here the comparative phenograms for your convenience. So accordingly, in case of treatment 5, 9085, and once again 9083 for the treatment 5. Accordingly, for the treatment 6, here we can see the difference, followed by this one, faint one, 9083. Once again, treatment 7, which is the most promising combination. Here we can see how much intensity is coming, and accordingly, once again, in the 9083, very faint. Now, once again, in the treatment 8, once again 9085, followed by 9083. So one thing is very clear from this phenomic study, that 9085 is having very high potential as far as nodulation is concerned, and the lowest one was observed in case of 9083. As far as tabulations are concerned, once again we can see here treatment 7. We can see for the 9085. So here is the highest mean value. Of course, its standard error is 0, but certainly mean value is very high. Accordingly, in case of 9083, once again, in case of type 7, once again high. So here one thing is very established: the combination T7 treatment 7 is very promising as far as agronomic perspective is concerned. And as far as genotypic perspective is concerned, certainly 9085 is the most promising genotype out of these 20 genotypes. So accordingly, this is another form of the presentation. Here once again we conclude that treatment 7 is the outperforming in case of 805, and accordingly once again 1083 is the outperforming in case of the treatment 7. So one thing is very clear that treatment 7 is the outperforming, and future prospects for these out of this study. What we could found that the genotype 9085 was the most promising, followed by the treatment combination SEBI, as well as CANFP, the gene which is supposed to be. Now this what is the prospects for harnessing these genes for the enhanced symbiotic nitrogen fixation in case of the chickpea. As we all know, the chickpea drop genome already available in case of chickpea. It opens new avenues for implying genome editing tools such as CRISPR-Cas system, targeting at the CANFP gene, which is very promising. Following, once it is done, these CANFP gene can be integrated with the promising genomic tools, whether this is the CRISPR or other Cas modifications of the CRISPR or other genome editing tools, because now there is a huge number of genetic engineering tools also, like zinc finger nucleases, transcription actuator like effector nucleases, which is also known as talents, and Cas system. Uh, these technologies enable precise genetic modifications, allowing the introduction, deletion, or alteration of specific traits linked to ergonomic performance. In particular, the CRISPR-Cas system is very simple, having high efficiency and versatile, and has revolutionized targeted gene editing right now, and holds immense promise. But what I mean to say here, this is not only CRISPR, because CRISPR has taken the pivotal seat, because it's comparatively easier and robust, and it has been proven in number of systems. But there is a scope or avenue to exploit other genome editing tools also. Anyway, future research can be focused for targeting these genes, particularly CANFP, associated with the nodulation and biological nitrogen fixation, to optimize symbiotic efficiency and improve overall crop yield. We propose to harness the strategy as advanced in tomato, as we have already postulated and published in 2023 in the Frontiers in Genome Editing. So that strategy can also be borrowed in case of chickpea also. So now let us have a brief idea about the genome editing tools, because it's not confined only to the CRISPR-Cas9. Site-specific site-specific nucleases, which are also known as SSN. These are having four types of genomic tools. One is JFNS, another is TALENS, third one is the endonuclease, and fourth one is the CRISPR. These are the genomic tools for which have, these are site-specific, and others are like site-specific recombinase, which are also known as SSR, and oligonucleotide directed mutagenesis, accordingly base oblique prime editors, which is the latest one, and this is more precise, and it has lots of potential still, but its implementation is not very easy. So in case of ODM is supposed to be the first genomic tool, which dates back to the early 1980s, is a gene editing tool that is a base pair specific, precise, and non-transgenic that has been greatly enhanced to the create unique and commercially relevant features in agriculturally important crops. It can be applied even in case of chickpea, but potential seems to be very less right now. ODM is a technique for targeted mutagenesis that implies a 20 to 100 base oligonucleotide, whose sequence is alike to target sequence in the genome, excluding a unit base pair change to achieve site specific it of the sequence of the interest. Applications are tremendous, like it can be utilized in case of chickpea, particularly for the herbicide tolerance, or biotics like heat drought, salinity, and biotic stresses like insect, bacterial, virus, as well as in case of nitrogen fixation, I am talking about nutritional enhancement and yield and other components. Other technique is the site-specific recombinant technique, which can manipulate genomes and stimulate or deactivate gene expression in numerous organisms. And these have already been demonstrated in the 90s as following. CRE logs from bacteriophages in case of E. coli, FLP, FRT from Saccharomyces cerevisiae, and RS from Zygosaccharide These enzymes catalyze recombination up to 34 base pair opposite to the odium in length in between its analogous combinations, side blocks FRT RS with an unequal core or spacer section flanks inverted repeats that act as recombinant binding locations. Once again, these can also be utilized for number of like identification along with the binding of recombinant dimers to mandatory sites, synaptic complex formation between bound position, strand exchange and fusion proceedings mediated by recombinase, synaptic complex segregation, and these can have different here in to simplify it just I'm presenting in the diagramming form. There are two strands and a crossover takes place and recombination. After recombination, here we can see this is a hybrid, this is a hybrid. Accordingly, crossover then accessory sites can be. So these are the two types SSR facilitated DNA strand in the first step and the second crossover output as here. So we can see that this technology can also be exploited in due course of the time. Accordingly, site-specific uh, SSR organization unforced that the majority of the 1,000 node site-specific recombination systems. Once again, only two families, tyrosine and recombinase, serine recombinase. Tyrosine recombinase interchange, disrupt, and rejoin two DNA strands in a holiday model. There are four-way connection intermediary in which two strands are non-recombinant while the remaining two are recombinant. In the experimental genetics and biotechnology, several tyrosine recombinases have been utilized. In fact, the most extensively used SSRs such as CRE, FLP, and RR members of this family. And once again, we can say this holiday model, like we can say here break is taking in only one strand and then crossing over is taking place. And here in case of the break is taking in two strands and we are getting the hybrids here. So there is a interchange in the two strands we can see. Now, serine recombinase cleave all four strands at one time in the is a synaptic complex, creating double strand breaks at the center of each carpuscle site. Breaks the DNA strand by the aggregation of the phosphodiester bond with the OH group of serino amino acid and covalently attaches the recombinant DNA to the 5' end at the breakdown. During recombination, serine recombinases create instantaneous double-strand breaks in both recombinant sites and there is no holiday junction. The serine recombinase family contains Phi-C31 integrase and Phi-C31 exogenase. So we can see here once again what we can see that these breaks are taking in the all four strands, and so once again in the different second set of the recombination we are seeing that all four strands have the interchange segments in each other. Another is the tool is the site-specific nucleases, which is also known as SSN. And site-specific nucleases are divided into following four categories like GFN, TALENs, meganucleases, and CRISPR systems. So once again, GFN are zinc fingers combined with the Hawk nuclear domains, which act as site responsive GFNs and cleave DNA in vitro in well-defined locations. The chimeric protein has a modular structure because each one zinc finger domain recognizes the form of a triplet. This approach was used to alter culture cells, including both model and non-model plants. So here we can see uh, GFN, what we can see, those we can have here, we are getting infected with the GFN, and after that, here we can have the hybrids or interchange segments, non-homology in joining, but here homology directed repair. So here we can say this is also one of the tools which can be utilized. Application, once again, by and large, application in case of all the tools are with little modification are uniform. So despite the challenges faced during the construction of JFRs, they have been widely used to modify genes of cultivated crops, particularly in the Arabidopsis, tobacco, maize, soybean, and canola. There is no utilization as far as chickpea is concerned so far. Accordingly, this has been utilized in case of soybean also, and it has creation of lines of chickpea with only two tranches have been described so far, but no output has come, which is stable. So now homing or meganucleases. Once again, this is one of the very potential area, but it needs to be worked upon. So, to make site-directed double-strand breaks in the genome, meganucleases, also known as homing endonucleases, are unique enzymes with high acuity and long recognition sequences, up to 14 base pairs that digest target DNA in a site-specific. But and it has a very high potential, but still not very successful. Accordingly, talents are easier to construct and more widely used than JFNs comparatively. But it has been used in case of the sweet 14 and producing mutants rice, so resistance in case of rice and knocked out by this approach so that it is responsible for pollen abortion and leads to cytoplasmic male static rice. It can be utilized in case of rice. So once again, activator and pair of talents, we can see how these alterations are taking place in case of rice for the somatic, sorry, this pollen grains. And application, once again, multifarious can be used to chickpea also for the heat tolerance, yield trace. As far as nitrogen fixation, I foresee it can be used, but so far no reports are available. CRISPR, of course, this is the very widely used technology across the globe right now. CRISPR technology was introduced 2 years later after the discovery of talent proteins. CRISPR, which consists of the non-coding RNAs and Cas proteins, was developed and has since become uh, widely employed. The most commonly used genome editing tools are TALENs and CRISPR associated Cas9. CRISPR that we associated with Cas9 is the most popularized and widely used across the globe right now. Each represents a type of the engineered nucleotide that can be customized to recognize, bind, and cleave a specific sequence in the genome. Unlike first generation genome editing approaches, CRISPR-9 Cas9 is easy to design, clone, and similar Cas9 protein theoretically can be used with a variety of the guide RNA to target several locations throughout the genome. Out of the three types of CRISPR, type 3 is used for plant genome editing. Two potential genes, 4CL playing important role in the uh, phenylpropanide metabolism and revel-7, RVE7 involved in circadian rhythm were chosen for CRISPR-Cas9. In case of chickpea protoplast thing, which is very responsible, has been already reported for the drought tolerance. Which is suppose the first report as far as chickpea is concerned, as far as genome editing is concerned. In chickpea protoplast plateauing, it is a revolutionary technique for accomplishing targeted pathogenesis. In comparison to the 4CL gene, the RVE7 gene showed excellence in vivo editing effectiveness. Gene CRISPR-Cas9, once again, this is a model which I'm already published, and it can be harnessed. How it can be harnessed in case of chickpea, we can see that how DNA cut is taking place, how trace RNA is being integrated, and how DNA repair mechanism takes place, and finally, non-homologous enjoining and directed repairing is taking place. So limitation and benefits of the CRISPR technology, of course, we can have multiple uh, benefits as well as some of the limitations also. Limitations can be like deficiency of CRISPR tools, immunotoxicity, target site are very limited, toxicity and immunological, immunogenicity of the crops in protein, and benefits of the CRISPRs are multifold, like high accuracy, simplicity, then cheap, high advantage, and cost effective, easy to use, disease cure, and no DNA, uh, no DNA something not readable. I am not able to read. So we can say there are multifarious benefits also. So, but whenever we use, as we say, every coin has two sides. So in isolation, it cannot give anything unless until we go for the very specific. So conventional building can be supported with the utilization of the genome editing tools. And once it is utilized in a very efficient and targeted way, certainly we can have better output or production in case of. So of course, I will cut short because I think my oration is getting up. The base and prime advanced approaches, once again, these are the very focused area and taking the pivotal place in the near future. And these have very high promising because these will be more precise and accurate. And but in the very infancy stage. So, base hitting, I'm not going to discuss much. Accordingly, prime hitting, I'm not going to discuss much because time is a limitation. So, but certainly we can say various genomic approaches, whether multi-omics, transcriptomics, proteomics, metabolomics, pan genome editing, etc., have tremendous potential to influence the plant breeding. Accordingly, the editing the target DNA sequences by adding, deleting, etc., can have utilizing the appropriate technology crop to crop or trait to trait, can have the immense potential. Of course, CRISPR-Cas9 right now it needs to be very speedy because this is the proven one, but other technologies also need to be standardized. So conclusion is that out of our studies, we conclude that these out of these rhizobium and BAM is the most effective combination, which from the ergonomic perspective, it can be utilized for enhancing the nodulation and overall productivity in case of the chickpea. Accordingly, the genotype 9085 can be utilized for more specified studies by the biotechnologists, plant breeder, and genome editors, etc., etc. So, to enhance the nodulation process in case of the chickpea, and with all these now the future prospects, as I said, combination rhizobium and BAM is a very effective one. CRISPR-Cas one again can be integrated with the gene. Already we have studied the uh, uh, impact. So CANFP genome can be gene can be edited through utilizing the CRISPR-Cas technology in case of other genotypes, integration of microbial treatments and genome editing once again. And finally, these are the three output out of my study, that the combination rhizobium plus BAM, genotype ICC 9085, and the gene CANFP. These are the output which can be harnessed for enhancing the nodulation and overall productivity of chickpea, and ultimately giving a good output of the productivity, total production and productivity in case of the pulses. With all these, there is a huge number of team. This is not my alone work, because almost entire country has contributed. ICSET, ICR, IRI, there is apports. Then ICSET, ICR, National Institute of Plant Biotechnology, Udaipur Agriculture University, Jammu Agriculture University, Palampur Agriculture University, SWAT Allahabad, Regional Station Pusa, IRI, then Central University Samastipur, Govind Vallabh Pant Agriculture University, Chandrasekhar Azad Agriculture University, Sardar Vallabhbhai Patel Agriculture University. And I am very happy to say that these other institutions have contributed only on the basis of interpersonal relations, and the vice chancellor of these all universities were kind enough to honor me, and they supported all expenses in their premises. So, with all these final acknowledgement to the IRI, and thank you all very much for giving me opportunity and bearing me, because my oration was a bit long. Thank you very much. Any question and clarification if required, please.
Thank you very much, sir. We have one question from Abhishek, who says that can we say that everything totally depends on bioinformatics?
No, everything cannot be depend on bioinformatics. Once again, as I told you, that these need to be integrated, and unless until our phenotypic data are robust, bioinformatics cannot contribute significantly. And I don't know why Abhishek is emphasizing only bioinformatics. We have five fingers. Each finger has equal role, unless until we integrate everything in a appropriate way, or optimized and standardized way, we cannot harness any better output. Certainly, bioinformatics cannot be ignored. It has to play a vital role, but it cannot be the sole criterion for total output.
Thank you very much, sir. We have a bunch of questions, but I am so sorry we are running out of time. We'll request everybody to please share your questions related to this topic, and we'll forward the same questions to Dr. Rajan Kumar, sir. You can share us your questions on our email ID conf.isgbbrd@gmail.com, and we'll revert back with your answers. Thank you very much, Dr. Rajan, sir.
Okay,
It
thank
was
you very much
wonderful
for
listening to you.
giving me opportunity. Okay, thank you.
Thank you, sir. Next in the sequence, I would like to invite, without wasting much of our time, Anbhuti ma'am. Madam will be speaking on the topic vegetable oil industry. Over to Anbhuti ma'am.
Thank you very much, Dr. Anshuman. Today I'm going to present. Am I audible?
Yes, ma'am, you are audible.
Doctor. Yeah. And the slides are also visible.
Yes, everything.
So today I'm going to present some overview regarding the vegetable oil industry, because vegetable oil is very important. Oil at consumption is the major consumption in our Indian scenario as the edible oil industry. So, I will give you some overview regarding the oils and fats production, consumption, and its impact on the economy of the Indian scenario, as well as the consumption taken up by the women, men, kids, and so on. So, first of all, I'll give you some overview regarding the oils and fats in world, in India, as well as other perspectives. So, if you look at the oils and fats production in world, the total production is 185 to 193 million tons per percent share. If you look at the percent share, you will find that it is the palm and palm kernel who is having the largest percentage share, and followed by the soybean, and later on it was the rapeseed mustard. So rapeseed mustard also belongs to the percentage share, maximum third percentage share in case of the oils and fats. So if you look at the oilseed productivity in India as well as the country versus country with highest productivity, so you will find in this case the groundnut tops the position. And groundnut, it is having the highest productivity in USA, whereas rapeseed mustard has the highest productivity in Germany. Of course, it is followed by various other countries also, including China as well as India also. But here we are talking about only the countries with highest productivity. And these are the other edible oils which are having highest productivity in different countries. And if you look at the castor oil, you will find it is India which tops the country, which tops the globe. So per capita consumption of oils and fats in major countries, that is very important, because that shares that whether we are going to import the oil or we are going to export the oil. So in this case, you will find that per capita consumption of oils and fats in USA, it was it was 35.4% earlier in 70s. Then in 2000 it was approximately 50%, and then in after 2010 it was increasing to 56%. And European countries, they are also having the highest percentage. But if you look at the Indian scenario, you will find that in 2010 it was lower than that of the 20%. So if you look at the compare position of India as compared to that of the EU, as USA and China, you will find it is in the better condition. So what is the world vegetable oil scenario? So if you look at this, the world vegetable oil scenario includes that palm oil is major exporter oil, that is approximately 46,600 lakhs per tons, and it is followed by that of the soybean, and then it was followed by the sunflower and rapeseed. So rapeseed oil comes at the fourth position, although rapeseed consumption is highest in case of the Indian scenario. So if you look at this palm oil, it accounts for 36% of the total vegetable oil production. So it is very important to maintain the in Indian economy. So if you look at the vegetable fats, you will find that vegetable fats and oils, they are going to affect the nutrients or energy. For nutritional and as well as the health professionals, they said that this oil is better, that oil is better for the consumption from the human body perspective, and so on. Similarly, it is also dependent upon the crop that can be converted to the commodity. So agriculture related personnel, they are going to affected by the vegetable oil percentage share, production, as well as area. When you go for the chemists chemical content or the biomolecule, again this is very important, because there are so many all of us knows clearly that vegetable oils and fats are the highest energy producing component in the body. So if you look at the nutritional requirement. Definitely all of us knows that most of the vitamins, they are oil dependent, or oil uh, required. In case of the dietary fat, now comes to the dietary fat, which is related with that of the nutritional perspective. So dietary fats includes, which is going to affect the structure or membrane composition of a cell. So if a cell is affected by the dietary fat, it means that dietary fat is going to affect the energy level of the body also. It also provides essential fatty acids, and it also affects the absorption of fat soluble vitamins. As I told you earlier also, that fat soluble vitamins are very important for the biochemical pathways present in the human body. So whatever we consume, it all depends upon the solubility of the vitamins coming from our uh, foods. Similarly, these vitamins which are fat soluble, they are also involved in the improvement of palatability of the diet, and metabolize they are directly metabolized into active lipid mediators, and they are going to affect the role in the metabolic diseases. They also affect the obesity, diabetes, and various other diseases. So that's why it is said that if you want to lower down the incidence of various diseases, dreaded diseases, in that case, we need to check our dietary fat or the oil consumption we are taking. So that is very important aspect. Similarly, the quantity and the quality of fat, that is again very important. If the quality of fat is like that, the shelf life of that oil is affected because of the oleic acid. So if oleic acid is high, in that case, that oil can be consumed for a longer time. But if oleic acid concentration is less in any of the fat, in that case, again, that oil cannot be used continuously for a longer time. And the quantity of the dietary fat is also important, depending upon the weight of the body as well as the internal metabolic rate of the body. So that dietary fat is very important. It has been seen that total fat from the diet should contribute at least 20 to 30% of the total energy or the calories required for the body. So this is a recommended daily dietary fat intake for Indians, like adult men and adult women. So if you look at this, that the total 20% of the total energy should be met by the fats, in which 10% should be the visible, and 10% should and the non-visible fats. And this is the recommended daily dietary fat intake for Indians in case of the children, boys, and girls. So it is the age group. On that basis, it can be said that the children of the 3 to 18 years, 25% of the total energy should be met by the fats, of which 15% is from visible and 10% from the non-visible fats. And for infants, up to 40 to 60% of the total energy should be met by the fats from human milk. So human milk consumption by the newborn baby is very important, because in that case, the 40 to 60% of the energy will be met by the fats. And for infant of the 7 to 24 months, it is said that 35% of the total energy should be met by the fats, from the 25% of the visible and 10% from the non-visible fats. So if you look at the oil consumption patterns in India, you will find that it is the palm oil which is taken up. This is the percentage consumption by the different by the Indian society for from different oils. So if you look at the palm oil, it is the 36%, followed by soya bean, then third was the rapeseed. Now this 10% is always variable. It all depends upon the location of the state, location of the district in the state, that that is going to be affected for the consumption pattern. So if you look at this, you will find that coconut, castor, solvent extracted oils, tree and forest origin, linseed, Niger, they are required in a very less amount. But Indian community likes soybean, mustard, sunflower, groundnut, and so on. And if you look at the various major exporting and importing countries of the edible oils, you will find that in case of the groundnut, China and Italy is the major countries, whereas the India's position is the fifth largest exporter. Whereas in case of rest of the oils like mustard oil, sunflower oil, soybean oil, you will find that it is the seventh largest in case of the mustard oil, but again in sunflower, soybean and palm oil, it is the largest importer. So we have to lower down this import condition because we are importing from China, Turkey, Netherlands, and so on. Right now in 2025, this table is now changed because in this case the palm oil production in India is increased tenfolds. So that this table has been changed right now. Now, this is the demand and supply of the vegetable oil. So, if you look at this, the value in crores, it was the in 2012-13, it was the total requirement was 54%. Now, in case of 17-18, again it was 59%. But in 2024-25, it is less than 40. It is less than 55%. Now, if you look at the net availability, import, and consumption of the edible oils in lakhs and tons, you will find in it was 267.10% of the net availability and the consumption. So, what is the edible oil industry in India? It is the one of the largest edible oil economies with oil mills, solvent extractions, vanaspati plants, refineries, along with more than 1 million people who are involved in this industry. So the size of this industry is approximately rupees 6 lakhs millions and import export trade is approximately 130 millions. So if you look at the India is the leading producer of oilseeds. It contributes approximately 8 to 10% of world's oilseed production, and it is the fifth largest producer of oilseed in the world behind US, China, Brazil, and Argentina, and it accounts for 7% of the total world oilseed output, 7% of the world oil meal production. That is again very important because India is the prime exporter for the oilseed meal to the world, and oil meal exporter, vegetable oil production, and so on. So if you look at this graph, it is clearly mentioned here that the production and the area is increasing, whereas the yield is decreasing slightly. So what is the major cause for this thing? Where area is there, where production is also there, although the increase in the production is not very fast, but even then the yield is low. So these are the major issues which is directly affecting the oilseed production, and that is first very important thing is that there is increase in production and the demand that is not increased. Demand is increasing, but production is not increasing in that percentage. That is one of the major constraint. Another very important constraint is that the production is largely in the rain-fed area, so it depends upon the rains, and you know that global warming has changed the rain pattern also. And third is very important that the production which is rain-fed condition, that is more prone to biotic and abiotic stresses. Again, that is very important aspect which is going to affect the oilseed industry. If you look at these graph, you will find that it is the crop-wise oil consumption pattern in India. So if you look at this, India, Delhi, Punjab, Jammu, Kashmir, Himachal Pradesh, Uttar Pradesh, they are mainly dependent upon the sunflower as well as the groundnut, palm oil, and now the pattern is changing towards the rapeseed mustard also. So it is this graph shows that this is the consumption pattern in the India which affects the oilseed industry also. So vegetable oil industry, it is significantly influenced by the effective byproduct utilization. Now, byproduct utilization is that seed is used for the oil production, but rest of the things like stem, like leaves, all these should be used for the value added product. So in that case, again, that there will be increase in the vegetable oil industry economic wise. So this sector is very important because it is directly affecting the Indian economy, and directly it is associated with the foreign exchanges like 90 million per annum. So it accounts for India, as I told you earlier also, that India accounts for 5% of the world's vegetable oil production. So these are the different methods by which oils can be extracted out, like from ghani, from expeller, from solvent. So if you look at the ghani, it is only 20 to 30% of the oil is extracted, but we go for the expeller, it is 34 to 37% of the oil will be extracted. But when you go for the solvent, it is 40 to 43%. But if you look at the quality wise, it is the ghani which is showing very good quality of the oil extracted, followed by the expeller, and followed by the solvent extracted. So this is the Indian edible oil structure, where you will find that in India the number of units for oil mills are approximately 15,000. For solvent extraction plants, that is 600 approximately. Vegetable oil refineries are approximately 465, and Vanaspati units are approximately 250. And the capacity for the production of oils that from the oil mills is very less, that is 20 to 30% only. So there is a need to increase the oil mills to the vendors, as well as to the farmers also, so that it will directly affect the to increase the farmers income, as well as the commodity increase will be there, because consumption is more in India. So this is the major oil industry, as I told you earlier also. So these are the some state-wise vegetable oil refinery industries in 2014, which has increased in 2014-24 by 10%. So if you look at the Andhra Pradesh, the refineries were more in Andhra Pradesh as compared to that of the Uttar Pradesh. But now the system is changed. Now even in Rajasthan, the refineries are increasing day by day. So on that basis, that there are the duty structure, as well as the exempt policy was developed to increase the oil percentage and to lower down the import, and to increase the export. So in that case, you will find that 30% exempt policy was given to the oil seeds, except the soybean, which is having 45%. So this is the Food Safety and Standards Act. According to 2006, it has been found that the manufacture, storage, distribution, sale, and import should be should follow the standards, Indian standards, for the availability, to ensure the availability of safe and wholesome food for human consumption. That is again very important to for the Indian policies. And these are the different regulation, where you will find it is the licensing, registration of food business, food product standards, laboratory sample analysis, prohibition restrictions, food and health supplements, nutraceuticals. These are the different food and safety regulations which are going to affect the import also, because if we are following the same regulations, then the export will increase and import will be lower down. So these are the standards, where you will find that standards can be divided into vertical and horizontal, and vertical will directly affecting the quality parameters, whereas horizontal is affecting the safety parameters, that is labeling, additives, pesticides, contaminants. And quality will affect the quality of raw material, ingredient, and quality of the finished product. For example, it is oil. So standard development process in case as per the FSSI is that scientific panel is there, scientific committee is there, food authority is there, legal vetting for the Hindi translation, and final notification in the Gazette of India. This is the flow chart for the standard development process by the FSSAI as well as by the BIS. So these are the different food safety additions for the sub-regulations of fats, oils, and fat emulsions. So these are the different sub-regulations. And refined oils, again, it has been said by the FSSAI that which are standardized, edible oils which are standardized under FSSAI may be refined, but rest of the other oils cannot be. So these are the different provisions which are related to the trans fats. Like trans fat limit in the interesterified vegetable fat should not be more than 5% by the weight. For example, in bakery and shortenings, and the exact quantity of the trans fat should be declared on the labels of the oils and fats and the foods. For example, if you are buying a cake, so they on that cake, the declaration note or the labeling should be like that the trans fat should be mentioned there, because trans fat is not good for the health, and it should be less than 7%, or as FSSI it has been said that it should not be more than 5%. So if the label shows that it is more than 7%, it means that it's not edible. So this is the labeling requirement that what is the name, nature of the edible vegetable oil, as well by percentage, by weights, and so on. So these are the recent initiatives to increase the export as well as to lower down the import. That edible oil, vegetable oil should have developed total polar compounds of 25% not to be used. That is very important. So trans fat again comes into picture, that it should not be more than 5%, and the virgin coconut oil, peanut oil should be mentioned as per the new standards. So this is the last slide, which where we can say that what is the way forward interventions and so on. So again, you will find that these are the, there should be increase in the oil extraction units, like self help group should be there, farmers should be, association should be activated, and KVK should also take some initiatives. And nutritional requirements of the vegetable oil per annum per person should be only 12 to 13 kilograms, but right now it is 19 to 20 kilogram per annum per capita. So it should be reduced to 15 kilograms, so that if we reduce from 20 kilograms to 15 kilogram, we can reduce directly 5 million tons of the import, which cost approximately 25,000 crores per year. So this much amount of we can add to the Indian economy. So this is the way forward interventions. Thank you very much.
Thank you very much, ma'am. It was wonderful listening you. And I guess now we are able to see the slide. Thank you, one. It was wonderful having you with us, madam. I hope we all enjoyed this session. If you have any questions, you can send them to us on our email ID, and we'll get them resolved through Dr. Anbhuti, madam. Thank you very much, ma'am, for joining in.
Thank you. Thank you.
Next, I would like to invite Professor Dr. P.S. Brahmanand, sir. Sir is the project director of Water Technology Development, ICAR-IARI. Professor Dr. Pothula Srinivasa Brahmanand is two-time Guinness World Record holder and agricultural scientist. He is presently working, as I told you, as Project Director, Water Technology Center, ICAR-IARI. Prior to this, he served as Director Research at RPCAU Pusa, Bihar. He is presently Chairperson of FAD 17, Farm Irrigation and Drainage Systems, Sectional Committee of Bureau of Indian Standards, and core group member of National Task Force on Integrated Water Resource Development and Management of Niti Aayog. Dr. Brahmanand received two international awards, Guinness World Record holder in longest speech marathon in 2012. His 33-hour and 46-minute speech on science and technology, society and human development included topics of agricultural sciences and drawn the world attention. Guinness World Record holder in fastest time to identify all elements of periodic table in 2016 in 3 minutes and 54 seconds. He also delivered longest extempore speech of 38 hours and 21 minutes on ideology and spiritual concept of Swami Vivekananda and its relevance to the present day society. In completely standing posture on the occasion of birth anniversary of Sri Ramakrishna Paramhansa during 18 to 20th February 2014. He has bestowed with five national awards, including Indira Gandhi Rajbhasha Puraskar of Ministry of Home Affairs, Government of India. He has received this award twice for original book writing by an Indian author in Hindi language for the year 2005 to 6 from former Union Home Minister Shri Shivraj Patil during 2007 and 2010 to 11 Honorable former President of India Shri Pranab Mukherjee during 2012. For authoring two original books in Hindi, ICR Award for Outstanding Interdisciplinary Team Research in Agriculture and Allied Sciences twice for Biennial 2013-14 and a lot more. He is also Fellow of Indian Water Resources Society IIT Roorkee and Andhra Pradesh Academy of Sciences. Dr. Brahmanand has published 103 research papers in reputed peer reviewed journals, including Nature Communications. He contributed immensely towards post flood management and index based flood insurance for farmers of Bihar and Odisha. Design and development and impact of ICR flexi rubber check dams, estimation of impact of ICR flexi rubber check dams on agriculture, methodology of watershed evaluation, state irrigation plans, and district irrigation plans, and a lot more to say about Dr. Brahmanand. I would like to add one more thing and would share with you all that Dr. Brahmanand is currently in Malaysia. He is there for a government event and he is joining us directly from Malaysia. Huge welcome, a warm welcome to Dr. Brahmanand, sir.
Thank you very much, Dr. Anshumanji, for giving me this opportunity, for inviting me. Respected Agarwal sir, Dr. R.C. Agarwal sir, Rajendra Kumar sir, and all other esteemed participants who have assembled here. First of all, good evening to all of you. I am presently from Kuala Lumpur, just with I.S.G.B.R.D conference. I'm just attending that, and in between trying to just allocate some time for this. First of all, thanks to the Indian Society of Genetics and Biotechnology Research and Development, and also St. John's College, Agra, who is organizing this important conference. And also thanks to Anshuman. Let me just go directly into the innovations for water conservation and management for ensuring food security. Here, the main aspect is we are aiming for making a sustainable intensification, wherein we are aiming from the crop improvement side, from the genetic side, from the plant breeding side. We are trying to match the the required potential which is required for the food security. But to make it happen, one of the most important aspect is from natural resource management, and especially from the soil and water conservation. So, keeping that in the mind, now I will be concentrating on what are those innovative technologies which can be really relied upon by the researchers, and which can help the farmers to really to provide the food security. So let me just see this one after another. Whether slides are moving, Dr. Mr. Ankuman. Let me see.
No, sir, you can switch on to the next slide.
Let me see.
You can click on slide 2, I guess, then it will move.
It's not moving now. Moving. It's not moving in this mode. Earlier, when you told, that time it's moving.
You can click on the next slide, sir. It will move, I feel like. Slide 2, if you click on that. From the sidebar, if you click on slide 2, it will move.
I know that, but the only thing is it's not coming. It's not moving. I told you, no, that time it was moving. You have checked that one. Now it is not moving.
Can you click on the slide 2 on the sidebar once? On slide 2, sir. Next to this one.
I know that, but it is not taking. That's what I'm telling. It's not when you're clicking now, it's not taking. I'm stopping this, and again I'm sharing. Let me share. Yeah, now it will move. Let me see. Yeah, is it moving now? Yeah.
Yes. Perfect.
Okay. Now, just this is a general phenomenon. Everybody knows that how the water it has its own way of circulation. Like out of the total precipitation that happens anywhere in the world, around 60% of that precipitation it will be utilized for the evapotranspiration. This is the slide which talks about it, and at different continents, how it is varying. This is showing about it. Let us first have a look at what is the status of water availability in India. If you convert the annual rainfall into the precipitation, in terms of BCM, it is coming 4,000 BCM for the total country, and the water resource potential is around 1,869 BCM. but everything is not utilizable. So out of that, only 1123 BCM are utilizable. The groundwater is 433 and surface water is 690. Now, out of the available water, now if you see in India, out of the total area of 141 million hectares of net sown area, the rain-fed area at this moment is contributing 45%, whereas the irrigation net irrigated area is contributing 55%. So out of that, again, if you see that if the farmers give the irrigation in the both the seasons, then it will be gross irrigated area, that is 120 million hectares. Some noise is coming. Can you mute it? Sorry.
Yes, sir.
So out of that, the micro irrigation, which includes the drip and sprinkler irrigation, they contribute around 16.7 million hectares. Now the issue is the per capita water resource, per capita water availability in India is coming down because of youth population, but the water resources are almost stagnant. So the issue is how to make a best utility, means water use efficient practices, how to develop it, and for that the innovations are required. We need to concentrate not only rain-fed and irrigated area, we should also see that how to utilize the wastewater which is being generated, especially in terms of the municipal wastewater. That is the depiction made here. And at the same time, for the irrigation, lot of competition from the other sectors like industry and energy. They are growing at a faster rate up to 2050, whereas the irrigation share will be coming down out of the total water demand. So that will enhance the pressure on the water resources. So we need to come up with some innovative practices. Now, at the same time, we also have to see the demand side management. This is the slide which talks about the predominant cropping systems, especially the green portion which you are finding here. There is a rice wheat system, and similarly, a lot of diversity of the cropping systems, because every crop requires the water in different way. Like sugarcane, rice, jute, they require huge amount of water, whereas the pulses, oilseeds, and millets, they require less amount of water. So, based on the cropping pattern, we have made an estimation of like total water requirement based on that, and we have for different states the different values, especially the UP automatically, because of sugarcane, rice in huge amount of area, the requirement is quite higher. Now at this moment the total world is concentrating on the glacier preservation, because the World Water Day, which we celebrated in 2025, that was on March 22nd. So when the theme of the this year's World Water Day is glacier preservation, because of the climate change, the lot of the rate of melting of glaciers has increased like anything, and because of that, the glacier the amount will be coming down, and at the same time, in short run, in the downhill region, the lakes which otherwise get the water from the glacier at an earlier rate, but now because of the higher melting, they are also causing the floods that we are calling as basically glacier outburst floods. GLOFs are happening. In Indian condition also, if you see, in case of Himalayan mountains, the states which are located there, they are having mainly the important glaciers. Siachen Glacier is the one of the important one, and the way in which these glaciers, they have shown the higher rate of melting in case of India, and I was talking about the GLOF and all has been mentioned here. We need to be more careful, friends, because in the world now, at this moment, we can say that Slovenia and Venezuela, they have all their glaciers have been completely lost. And earlier they were having good amount of glaciers, but they have been lost. So now in India also, especially in the Himalayan region, the rivers like Ganga, Brahmaputra, who are getting the perennial nature of the water flow, they're getting affected because of going to be affected because of the lesser volume of glaciers in the future, because the higher rate of melting that is happening. So we need to come up with some innovative practices, especially the soil and water management practices, which can reduce the impact of the climate change, which can reduce the impact on the rate of melting of the glaciers. So how to accelerate the change to solve the water crisis? There are some issues which we need to address. The one of the important issue is there is a lot of variation in the irrigation percentage of different crops. Similarly, among the states also, the only 5-6 states, they are having higher amount, more than 80% of the micro irrigation coverage, whereas the other states, they need to improve in terms of the area. So similarly, we also have to give a lot of stress on the in situ moisture conservation, because in India the precipitation is not an issue. The issue is the variation, and also we are not able to catch it. We are not able to store it. So government of India has come up with a slogan like, you conserve that, catch the rain wherever it falls and whenever it falls. So this is very important. Similarly, the wetland preservation is also important. River rejuvenation is important. So all these things with the in-situ moisture conservation, how we can do. Here we can have a look at the number of water bodies. This is the first census of water bodies, which was recently conducted by Government of India. For our just an idea, we are having around 24.24 lakhs water bodies in the country. They have different distribution in different states. And out of those water bodies, the ponds are the highest in number, followed by the tanks and reservoirs. Now, Government of India, it targets the storage of water. The per capita storage is very less. When you talk about the water conservation and water storage, the per capita water storage needs to be enhanced. The India's per capita water storage is quite lower, whereas in terms of relative comparison with China and other countries, it's quite lower. So we need to enhance it. This is under Amrit Sarovar Yojana. The Honorable Prime Minister in 2022, he declared this Yojana, and under that, the around 50,000 Amrit Sarovars have been targeted. But India has already achieved 68,000 Amrit Sarovars by the end of 2024. So this is the one where the picture which you are seeing is from the POSA Institute IARI of New Delhi, and which has been done with the design of WTC, Water Technology Center. And similarly, in case of villages also, generally the village ponds, the community ponds, which is under control of Gram Panchayat, the revival of those village ponds will lead to the additional water productivity, and especially the additional yield, and also the additional area under the crops, like cropping intensity also can enhance, and it will also solve the water quality problem also. And this was done in Haryana. Just I'm going fast because of the limitation of the time. Similarly, another very important aspect of soil and water conservation, especially in watersheds, is the tank and well system. If you construct a tank and well within the space of 100 to 300 meters, so the pair of this tank and well will complement each other. And set of those 15 tanks and wells, they will have a catchment of around 500 hectares that can irrigate around 60 hectares. And this is a very well proven technology, innovative technology. Similarly, this is another one. This is called the two-stage rainwater conservation technique, especially for the eastern India, where 1/10 of the total area, the farmer can convert it into the water body that's called refuse, where the fish will be concentrating up to the month of August. And after that, in the lowland, up to that particular period, the fish will move to the lowland paddy because their water level will be up to even around 1 meter. Whereas after the August, it will be completely restricting to the refuse. And here, from the fly to fingerling stage, the farmer will get the additional return. And this is a very innovative technology. It's called ICR Flexi Rubber Check Dam. The advantage is compared to the concrete check dams, the rubber dam, it will have a double folded rubber where the farmer can fill the water with the inlet pipe, and it will raise to the height of 1.5 meters, and it acts as a reservoir. And the series of these rubber dams will help the farmers in conserving more water and also soil. Here, the advantage is the life of the rubber dam is around 25 years. It was tested in IIT Mumbai with the hydraulic lab. And whereas in case of regular check dams, the cracks will develop. And here, another advantage is flexibility also. The number of crops can be taken more higher than the existing one in the several watersheds. And this is a very important technology. Similarly, now government is also concentrating on reducing the conveyance losses through the pipe irrigation. And Ministry of Jal Shakti, it has given requisition for developing standards for the pressurized pipe irrigation system, for which WTC is providing now. And this is now when we talk about the micro irrigation to enhance the water use efficiency, whether it is a source from the canal water or from the groundwater, how to utilize that water for the important crops, including even millets. Recently, in the International Millets Conference, it has been decided from the 15% of the irrigated area, we can enhance up to 25 to 30%. Especially the micro irrigation can play a huge role in this. The director of Indian Institute of Millets Research, when he visited the fields of the IRI research field, this is from the Water Technology Center. Especially this technology is integrated drip cum mulch technology. It conserves both the especially the water conservation and in situ water conservation, and at the same time weed control also. We also tested for the DSR, the direct seeded rice. Also, it is coming up very well in terms of water saving. And along with the sorghum and pearl millet, you can also go for in between. You can also go for the pyra cropping with crops like pea, and that has also come up very well. Even for baby corn, also it has come up very well. Now the total world is moving towards automation. So here also, wherever possible, not to all the small farmers, but there are several farmers who can really who are looking forward for automation. For that also, we have technology innovation is there. And this is one innovative practice which has been developed by Water Technology Center, where the farmer who is using the surface irrigation, in case of rice and wheat, he can utilize this one. This is a automated surface irrigation system where the soil moisture sensor in the field, it will signal whenever the water is required. When the plant is about to start its moisture stress, it will give the signal. Then the gate will open, and from the field channel the water will enter. And after the saturation is reached, again the reverse signal will come to the controlling unit. Again the farmer can close it, so it will save minimum 25% of the water. And this is also connected to the mobile, and farmers can easily do that. And similarly, we are talking about the protected cultivation, utilizing less water resources. How can we just go for it? These are some of the studies from Precision Farming Center from WTC. And this is from the protected cultivation. Some of the crops like tomato, then capsicum, they all can be done. And now there are some issues related to the coastal ecosystem, especially when the flood happens, when the water logging will be there, how to come up with some of the alternate practices. Here the picture shows like, over-aged seedlings which can have a better resilience in the flood conditions. Similarly, biodrainage with the casuarina plantation. Then the structure which you are finding here is the subsurface water harvesting structure in the coastal areas. If you go only up to a limited height, then the fresh water will come. Otherwise, if you go deeper, then the salt water will come. So that design also has been made by us. And similarly, some of the crops like water chestnut, macana, they also can be cultivated. So some of the farmers who are having risk to the flood, for them, for providing the insurance, government of India is having Pradhan Mantri Fasal Bima Yojana. We have provided technical support by coming up with an index based on which the farmers of Muzaffarpur district of Bihar, they have been provided insurance for 3 years based on the satellitic images. We have made the estimation of depth and duration of submergence, and the farmer who got more affected, he got the more insurance, and otherwise proportionately the amount has been paid with more accuracy. So India is having a lot of diversity. Some of the hilly regions like Himalayan ecosystem, where this is the picture which you are seeing from the Ladakh, when you can find some of the protected cultivation structures which have been made with the local resources, especially the soil which is available there, and which is also some of the materials which has already been used. And those materials, by making the recycling of that, how we can really grow some of the high value crops inside those structures. So that has also been done. And this is the way in which the capacity building and awareness can be created by showing the inline and online dripper system, and also the rain gun to the farming community. And this is the one in the ICR Foundation Day when Honorable Minister has visited. This is very important one. This is a 100 plant model from the Center for Protected Cultivation and Technology, which I am also looking after now in addition to Water Technology Center. This 100 plant model, the advantage is the nutrient solution will be provided with 100 liters solution. Within 45 days, the cycle will be completed. 3 different tyres will be there. 3 different crops can be grown. And in a year, it can be at least 7-8 cycles can be done. The cost was initially 40,000. Now we have made it to almost 26,000. The advantage is even the space wise also it takes lesser space. Even in the urban setups, when the youth they wants to take it as a entrepreneurship for the entrepreneurship, even in the urban setup in balconies of apartments also this can be fitted. So how to utilize the less space with a less cost, and how to provide employment to youth, and that was the objective of this one. So I have shown some of the technologies, innovative technologies which can help in the rain-fed areas for enhancing the storage, and also helping the policy like Amrit Sarovar Yojana, and at the same time in irrigated sector to enhance the water use efficiency like integrated drip cum mulch and also automated surface irrigation system, which has been awarded with the International Telecommunication Union recently in Geneva. This is a specialized agency of United Nations. This technology has been awarded. Now, combination of that water conservation and storage in rain-fed areas, and also developing water use efficient practices, including the precision farming, precision irrigation management in the irrigated commands. And in addition, we also have a technology called Jalopchar, that is a wastewater remediation technology. And because a lot of urbanization is happening now, by 2047, India will be having 51% of the urban population out of the total population. So that urban municipal wastewater is going to be generated. That needs to be treated and tested, and then if it is found suitable, then automatically that can be utilized for the irrigating some of the agricultural crops. So that judicious use of water resources is the most important one for the sustainable management, and especially the soil and water conservation technologies. The innovative technologies are there. Now, how to upscale it to the farmer is the most important thing, so which can ensure the food security, thereby ensuring the Viksit Bharat. Thank you very much for giving me this opportunity and an opportunity to share my thoughts here. Thank you, Anshumanji. Thank you very much.
Thank you very much, sir. It was wonderful listening to you. And I think that we have got many insights about all the things happening regarding water conservation and water. I see one question popping up here, which we received some time back, but I feel it more relevant to ask you now. The question is from Ms. Aparajita Singh, and she asked, as floods wash away fertile topsoil, how can technology and breeding and genetics improve root secretions or soil plant microbe interactions to restore the conditions after floods? So I guess this is the question most suitable to ask you, sir, and I wish to listen from you regarding this.
Yeah, thank you. Actually, this is having more of a link with the microclimate, especially at the rhizosphere zone. The issue is the flood is a very big issue. Flood is not a small thing. Actually, we know recently the way in which it has created havoc, and every year it is being, even in US or any country, now flood is a big issue. Now the point is when heavy downpour happens and along with big intensity of flow, it will definitely cause the soil erosion. And once that happens, we are mainly concentrating on the post flood management, because you can't stop it at that moment. So we are coming up with some contingency plans for the flood management, along with the supply of input material, because several times the plants will go, input material will not be helpful. So I am talking in general first that component. Then the second component is how the farmer can be really helped. The farmer can be helped by making him ready to face the post flood scenario. So in the post flood scenario, we are trying to provide them the material in time, how to maintain the community nursery, especially identifying the severely flood prone areas. In those severely flood prone areas, what we are suggesting, the farmers can go for the community nursery, so that when the flood happens in one of the block, where this nursery material can be supplied immediately. Now, this is what I am talking about, a macro management. See, we are now, this question is related to after the, everything is washed away. This is talking about rhizosphere management. So to talk about the rhizosphere management, the flood is a very bigger platform. It's not easy. First of all, making the plant to survive itself is becoming difficult. So what I consider here is, this is not only related to the flood. This question is mainly for the soil plant microbe interactions. The restoration is a broader question. It's not directly related to the flood, because flood is directly washing away the plant itself. So where is the question of the microbe interactions and all? Now we can concentrate on that in the post-flood environment, for which we are having the plans for the post-flood management. What we are doing, suppose if the flood is coming in the very beginning of the phase, like in the first fortnight of, in the first 30 days of the, Kharif season, then the farmer will still have the time for the getting the precipitation in the season. Then you can go for, like in eastern India, like West Bengal, Bihar, Odisha, you can further go for the short duration varieties. If it is happening in the mid phase, he cannot go for the again rice crop. He has to go for the other crops, like either oil seeds and other crops, or even some of the crops like maize and all. Now, if it is happening at the end, what we are suggesting is going for the early sowing of the rabi crop, including the some of the vegetables. So such type of post flood management, along with that, you can also club these type of micro management like rhizosphere and all that. So this question is mostly related to what I can say, like a general one rather than for the flood. So thank you.
Right, sir. Thank you very much for your great thoughts on this subject. I feel like that many of our young fellows got two insights about water, and people who feel like that water technology is not actually a technology, or it is just something very, very normal. I feel like they must have got a lot much to learn today. And I believe that many of our students from St. John's and other places who have joined this webinar will be definitely reaching out to us for more questions to ask you. And I feel rest assured that you will be disturbed a lot in this regard, because all our students
Yeah,
will just
thank
pop
you
up.
very much. I also would like to thank Upajay ji also. I think we met last time. I think he's also here. So
Yeah.
thank you.
Thank you very much, sir. It was great listening to you. Now, I would like to invite Professor Dr. Shokor bin Abd Razak from Malaysia. Professor Dr. Abdul Shokor bin Abd Razak is an associate fellow of Bioaromatic Center, faculty of CET, Perseirin Tun Khalil Yaqub, Kuantan Pahang Darul Makmum, Malaysia. Over to Dr. Abdul Shokor. And immediately after Shokor, we'll have our most special guest speaker for today. And the person who we are waiting all to listen is Professor Dr. A.K.S. Rawat, sir. So over to Dr. Shokor now. Your presentation is visible, Dr. Shokor, and we shall start. Your mic is on mute.
Okay. Assalamualaikum and very good morning. Is it very good morning there or
No,
afternoon?
it's
Very good
evening.
afternoon.
It's evening here.
Oh, evening. All right. Very good evening to all of you. Okay, my name Associate Prof. Dr. Abdul Shukur Abdul from Associate Fellow of Center of Bioaromatic, Faculty of Civil Engineering Technology, University Malaysia, Pahang, Malaysia. Okay, today I would like to share with a lot of you about my project entitled Food Security Animal Feed from from solid waste and potential plant, especially Tricantera giganta, or in Malaysia, local name we call it ketumayam, Indigo vera species, and Hibiscus cannabinus, or in Malaysia, local name we call it kenaf, okay, with high crude protein for broiler and free-range chickens. Okay. Okay, background study. This is the main issue, especially for food security project in Malaysia, and I done this under project national grant. Okay, so with the what called the raw material increase, and at the end product of animal feed, especially for broiler, is increase. It's difficult to farmers, okay, to cater it, and that's why this research we need to find out the local plant and to reduce the price in the market. So our objective is to develop animal feed from agriculture waste that can reduce dependency on imported ingredient. And second, to produce animal feed formulation by using the mixture of palm kernel cake, palm acid oil, Tricantera gantata, okay, Indigo fera species, and also now is hibiscus cannabinus. And third objective is to investigate the growth rate of chicken towards the formulation chicken feed, which we find the best FCR food conversion ratio to ensure that the chicken is available and is suitable for the market. This is as a simple methodology. Okay, in this presentation, I will show you two as a formula within we using the PKC and ketum ayam, and the second one is ketum ayam and hibiscus cannabinus. So during the study, okay, this is a short one because I've given about 10 minutes to present the outcome of this project. Okay, so we can show that the potential, huge potential of using the ketum ayam and solid waste within 42 days. Okay, we can find out that we can reach 2 kilogram. Okay, 2 kilogram with using the what called the ketum ayam and solid waste. But and then the advantage for using this raw material is the utilization of the agro-industrial waste. Second, from waste to wealth, we try to find out from waste to wealth. And third, environmentally friendly product and economically affordable price for the farmers. So now is the second one with a mixture of ketum ayam, Tricantera gagata, and hibiscus cannabinus. To show that Tricantera gagata and hibiscus cannabinus mixture as an alternative protein source to replace the commercial poultry to traditional feed ingredient. Second, traditional chicken feeding to environmental concerns. That's meaning that we try to use the solid waste as a potential raw material for replace the imported raw material. So this is for fulfill the sustainable development goals for the cluster number 12, responsible consumption and production. So research background, okay, as I mentioned just now, ketum ayam is a normal practice in the traditional farmers in Malaysia, but in commercial wise, it's not produced yet. But we try to make sure that we can get the best formulation from ketum ayam, indigo fera, and also hibiscus cannabinus. So which mean the ketum ayam have a protein, vitamin, and antioxidant. Meanwhile, for the kenaf or hibiscus cannabinus, this contain various bioactive compound that can benefit animal health, such as protein, fiber, and mineral. So problem statement, yeah, in Malaysia, high demand for chicken feed and global demand puts pressure on poultry farmers to find sufficient and cost-effective feed sources. And second, a need for sustainable alternative, and that's why environmental friendly product is most important, cost-effective, and capable of meeting the nutritional requirement of poultry is needed. And third, for environmental concerns, so traditional chicken feed contribute to unsustainable agricultural practice that can harm to the environment. So we try to change to from the traditional one to into the sophisticated, which mean has a high technology in this project. So for this project, cannabinus, hybrid cannabinus, and Triangantera gangata, so to investigate the donation as as the first one, okay, nutritional composition of Triangulata gangata and hibiscus cannabis in poultry diet. And second, to determine the sustainable production practice as a livestock feed for Triangulata gangata and hibiscus cannabis. Of course, scope of work, okay, for this study, okay, first assess nutritional composition, and we investigate the nutrient content of this plant and to determine their suitability as chicken feed ingredient. And second, feeding trial, okay, to see the growth rate of the DOC. Again, the implement feeding trial to assess the impact of this plant and overall poultry health. And the third, environmental impact assessment, analyze the environmental benefit of using this plant compared to conventional feed crop. So significance of this study for our project is the first one to especially this study is as I mentioned just now, is for food security, especially for the farmers. Okay, B40 and not 40-20, but for B40 because they it's difficult to find the what called the best price, the affordable price in the market. So about about 50 kg in Malaysia is about 140 Malaysian ringgit to 150 ringgit. 50 ringgit per 50 kg. It's quite difficult to the farmer, the small farmer, okay, and then for the SME industry to cater it. That's why we try to give the contribution to the SDG number one. And then for the second one is for the zero hunger, sustainable poultry farming can provide livelihood for community, helping to elevate the poverty. And second one, life on land, improve soil health by enhance the ecosystem resilient and reduce reliance on chemical fertilizer. So this is a literature review. Okay, I just skip it. Okay, so this is a methodology for hibiscus cannabis and Gangata. Okay, we do the preliminary study and we prepare the sample preparation and pelletize. Okay, the formulation we have about now is about 10 formulation, but cannot disclose here because this is confidential. But again, I give about three diet, the best diet in using this raw material. And then we do the nutritional analysis, okay, as per approximate analysis, and then press experimental setup. So we see the growth rate within 30 to 42 days, okay. Now for this, the second formula, we found that the combination or the mixture of hibiscus cannabis and Taranantha gancata have a potential to give the best diet for this project. Okay, from this study, it can show that Tricantera gancata and hibiscus cannabis are expected to provide significant level of nutrient, 25% and maybe more, meeting of exceeded the requirement for chicken growth and productivity. Second, okay, using Tricantera gancata and hibiscus cannabis could lower the environmental footprint of chicken production by reducing reliance on conventional feed ingredient. As a conclusion for this project, so it shows that the palm oil waste characteristic in palm kernel cake, palm acid oil, and Triganthera gangata, indigofera species, and hibiscus cannabis have been proven a high in nutrient that can fulfill the animal feed requirement. And second, the formulated chicken feed by using PKC, PAO, Triganthera gangata, hibiscus cannabis, and also indigofera species have proven have growth rate of chicken with food conversion ratio is almost within 1.4 to 1.5 and control the agro-industrial waste. So we choose the diet number 2 shows the best result for the growth rate of chicken toward the formulation chicken feed. That's all for my presentation, and I open to the Q&A session. I pass to the Prof. Dr. Ashuman, please. Thank you.
Thank you very much, Dr. Shokor. It was wonderful listening to you. I would request, I see Professor Dr. Samuel sir has also joined, and Professor Dr. Manjula R. Thomas madam has also joined in. I would request if they would like to share some thing on this talk. So your mic is on mute.
Hello, good evening. Is it okay?
Yes, yes. Okay, doc.
Yeah. So, Professor, it was really a good talk delivered by you. My very simple query is this: how can these two plants be used for phytoremediation? As I've seen during while you were talking about that, they have very good impact in soil fertility to improve the soil fertility. So, are they able to reclaim the soil? Have they any quality to reduce the impact of heavy metals, something like this? Can you give an insight on this also?
Thank you very much for your question. This is regarding the phytoremediation. So, is it using the phytoremediation using aquatic plant or what call in the land plant? So, when we talking about the phytoremediation technology, so I in my research, this is my PhD. I will share with you if we integrate with the phytorenovation technology and biorenovation technology in any phase within in industrial wastewater or municipal wastewater is what called it is municipal waste. It shows that the potential of phytoremediation to remove the contaminant for the domestic and industrial wastewater, meaning that if we're talking about the soil, that is shows that potential to using the phytoremediation technology to any place. For your info, if we use the phytoremediation technology, because when the nutrient in the water, okay, in the wastewater or the soil is high and capability the plant to uptake the contaminant from that the medium of what called place that it can remove, okay, is about 70 to 90% above the contaminant. And second one, when that area is huge with the nutrient, okay, the plant grows is very good. So you must make it harvest trimming the plant to make sure the aesthetic value is very beautiful. So when you trimming the plant you can process, okay, we can use for the raw material, especially for animal feed. It's not only for broiler, it's not only for free range chicken, it can use especially for the ruminant uh, what call ruminant animal. Yeah, doctor. That's my answer. I will. That's my opinion based on the experience for the vitamination and bioremediation.
Right. I hope
Yes,
Samuel
I
sir, he
answer the
was
question, doctor.
Yeah, there was some disturbance in my laptop. So it's good since they have very good roots. The root is very, they go to very good depth also. So that I was thinking that can be used as phytoremediation also. Thank you so much, sir, for your answer.
Yes, yeah, yeah. Thank you. My pleasure.
Thank
Any
you very much.
question on the day?
Thank you very much, Dr. Shokor. And
Yes, you're welcome.
without
Thank you very much to the secretary to invite me as a lead speaker for this international conference. Thank you very
it was
much.
a pleasure. It's a national conference, and since we had a conversation long back, so you are one of our only international speaker. Though Professor Dr. Berg was also supposed to join, but he had some engagement, so he could not join. But anyways, it was nice having you, Dr. Shokor. We shall now invite Dr. A.K.S. Rawat sir to talk about medicinal ethnobotanical plants. Dr. Rawat is the executive director in Manipur, and without wasting much of the time, I'll directly call on Dr. Rawat sir to speak on the topic, as we are running late of time, but because his introduction is very detailed, and if I speak, it will definitely take around 5 to 10 minutes for me to justify what has been mentioned in his brief. So I am so sorry, sir. I am not taking your brief, but your name is just enough. Also, I would like to tell everybody who has joined in that you all must have heard about one medicine, BGR 19. I am not sure about the name, but it is BGR 31. That has been launched and introduced by Dr. Rawat sir only. So over to you, sir. Warm welcome. So your mic is on mute, sir. So you're not audible. Your mic is on mute. You are not audible, sir. There is some technical glitch because of which Dr. Rawat was unable to unmute himself.
Acha.
Yeah,
Oh, I
perfect,
was
sir.
not
Now you're
aware of
audible.
it. So now it's okay, Anshuman?
Yes, perfect, sir.
Yeah.
Perfect.
Sorry for this technical difficulties. So, good
Not a
evening
problem.
to everyone. And at the outset, I thanks to all the organizers of this national seminar on fifth national conference on innovation in plant sciences, genetics and technology and sustainability futures. And I thanks to Dr. Upadhyay, Dr. Arvind Kumarji, Dr. Samuel, and all the office bearer of this seminar. So, as you know, uh, directly I go to my presentation. As you know, we are having technology national conference, innovation, plant sciences. If you see technology, if you see generally in these decades, we have a bioscience, biotechnology, we have information technology. Now it is we have another area we can say it herbal technology. So how we can utilize our plant wealth which we have in India? What are the opportunity to develop new combination, new drugs, new molecules, and from its botanical sources and our traditional knowledge also. And how if we have the leads from the plants with the from folklore, from traditional and from knowledge like we have the Ayurveda, Yunan Siddha, we can go and develop number of new combination, new molecules also, and thus we can support our masses for the healthcare or wellness also. And also, I will speak few words about the quality and of the importance of quality in this program. Also, next slide is I will just cover introduction, Harvard product development, and how many groups of drugs in India we have, and what are the rules and caution act, drug evolution, isolation, and some example. So quickly, I will go to the how we can utilize our information which is available in
tribals population, it is available our ethnobotanical knowledge. Ethnobotanical means the information which are available with the travel people. So we can say कि if you see 2011 census data show that more than 8,000 plant species are being used by the tribal people for their healthcare, for their area, I mean for the healthcare also, for nutrition also, and different purposes. And these are things, and more than 2,500, 25,000 formation are available in the ethnic, in the travel population all over the India, I am saying. So there is a great information which we can use for our drug development also. This is one example I will tell you. Just I told you 8,000 plant species are being used by the our tribals people. So we can say these are all tribal medicine. And similarly, we have folk medicine also. Folklore means the villages. In villages, what are the plants they are using for their different purposes, for eradication, for prevention of diseases, for curation of diseases. So we already data is there. 5,000 species are being used by these population villagers. And if you see our traditional, I mean, system medicine like Ayurveda, Unani Siddha. So we have a Ayurveda United Siddha means we have a Siddha also where 800 plants are being used. In Ayurveda we have more than you can say more than 1200 species are being used. So Ayurveda, Siddha, Unani also, and some modern medicine 30 plants are being used. This is about only for India. What we have 17,500 plant species, flowering plant, I'm saying, only flowering angiosperm, which are already recognized having medicinal value. So there is a great wealth is available about our information. And as we know, we Ayurveda, we have Charak Samhita, we have number of books available, and it is day back to 600 BC. You can imagine that time people know India Ayurveda about the Ayurveda. They know about surgery also. So that is a great achievement we have. And same time, 16th century to 17th century, you can see the in Europe also we have the number of people who are Greek and other area of Europe. They are also exploring the plant, local plant for their health care system. So, so it means India has a health wealth. We have the Rigveda also, as I told. Rigveda has Ayurveda also. In this book, Ayurveda is there. It's 4,500 to 16 BC. So it has mentioned that time 67 plant only. Today we have more than 7,500, roughly 8,000 plant species. But that time it was documented 67. And whatever the and Charak Samhita had more than 1100 plants are at that time. So there is a lot of scope to go through it and develop this our uh, drug which has maybe herbal sector. As we know the growth is very fast, 7%, more than 7% annual growth is there. And if you see by 20 already we have the data on that 5 trillion US dollar worth of global market is there, and India is hardly 2.4 billion US dollar. So we have, although I'm not saying only 2.5% of global market we export this much also, and we have 5,000 crores worth of rupees for I mean product we developed in India, and we have more than 900 manufacturers available in India also. So we have trying our best to explore this area for for the utilization of plants for medicinal purpose, for aromatic purpose, for nutraceutical also. These things we are working on that. So, as you know, ethnobotanical and traditional medicine, ethnobotanical information is available. For example, we have one plant, Vasaka. Vasaka is a very common plant, Aeroda vasica. And we have people who are working in this area, ethnobotany. They have documented so many things, plants which are being used by the local people. So, Aeroda vasica is cough and cold, even rheumatism also. They have reported that. And on this basis, we can directly go for the product development. So this data is there, and just example is there. We have the Wasaka leaf is here, and these are the compound. By doing research on this, we identify so many compound Wasaka leaf. They are all bronchodilator. They help in for our in cough and cold also. But information available in the lab, I mean, say hundreds of years. I mean, knowledge in the it's about the local people are being used for the cough and cold. So directly we can make the formulation of that. We can make tablet, whatever their syrup also. So the information how we are utilizing nowadays, although there is a big gap how to develop it, there is a rule regulation of that. That will I will come to later on. And quickly I will give you some example like Brahmi. We know the Brahmi, Bacopa, Centilla acetica, and Bacopa monterey also. And this is well, it is very commonly used in different formation tablet for brain, for increase your memory, IQ. And when people have work on like these are the work carried by uh, the Nalini and Aurora, they find that it has the aqueous extract is helping in learning and memory. So now scientifically we are also validating the thing. So I mean to say the number example like Texas Valenciana, it has been used now for the anti-cancer people, like tumor. For Taxol, Taxol is Taxol is a drug which is being used for leukemic cancer also anti-tumor agent. So we have already proved, scientists have proved it, but information available in the internet. It's easy to have a information and you do the scientific validation on that. Similarly, we have the aconite also, very good plant which are being used in homeopathy also. So it find that it is a antibiotic also and anti-inflammatory also. So like this, we have big data on that. Even one example like CDRI, we have one product on liver infection, Picorella curva, very good, important plant in Himalaya. And picoline is a compound, which is a drug which has been developed by CDRI, but they know the plant has been used for liver cirrhosis or something good for increase the your hungriness, appetite like this. So they take directly lead and make the isolation and they develop the new drug. So that part I will come later on. So just I want to know that there are number of plants, hundreds of plants which are already documented and we have utilizing for the as a herbal medicine or making a modern medicine or identifying some compound also, molecule. Similarly for the color also, we can go directly color. Color is natural color. So this is a very good area where we are working for the last 10 years. We are working also with an herbal dye for hair, color, face from the haldi also. There are number of RDBI novelis also. Number of plant we have Baheda, cinnamon, number of baths are available. Very good wealth are available to to isolate Indophila also to identify the color and utilize this. So they are big scope. We have done number of plant we have developed at NBRI when I was there and with the CMAP also. All the color lipstick which we give to Lip Mount Himalaya also. We have different colors from one plant. Single plant you can have hundreds of color combination, and we have patent on this also, and we make the this gulal during the holy festival also. So this is a good. I mean, just I want to tell you not only medicine, but color also we can have, which is having no side effect, even protective. They are antioxidant, so they are good for our health. Similarly, for the nutraceutical point of view also, one example very good nowadays, very popular also, moringa. Moringa sage. We find it's a golden tree we are saying because it has what not calcium four times than more than milk. It has vitamin A, it has potassium, vitamin C, protein. All the macronutrients and protein are available. So now number of company only they have directly they are making powder or tablet also and supplying this to the market also. The Himalaya also this is our EcoHerb. This is one company where I am working also. So we have developing tablet form also. So this is very good plant for the rural area also. They can use directly leaf and with the they can use leaf also making powder, and they can put 1 spoon in their in roti also, chapati, whatever they are having, and even sabji and vegetable also. So that is how you have to. We are exploring what plants which are nutritionally important. A number of plants are here, but just I am giving you one example, and how now it is big industry are here. You can see number of plant in Europe also. They are $100 of 60 capsule of this. This is so important nowadays. We are exporting this plant drug to USSR also. So this century, 21st century, we have rich biodiversity, and traditional knowledge is there, automotive knowledge is there, and science is there, modern. Only thing we have to convert herbal medicine, pharmaceutical, nutraceutical, and cosmeceutical, just to take the to healthcare popular masses, our public, and we can export also for increase our economic wealth, and same time give the employment over science graduate, pharmacists people, all the chemists, biologists also, and also other labor also in production. We can use all these. I mean, and their manpower we can use for their employment also. So there is a big chances to have this. Now, just in a short, because time is limited, two philosophy are there, two principles are there. One is you just screen the plant, whatever you have, identify X, Y, Z, then you test it on animal. If X compound X extract is good, then you can do later on to animal model, and directly you can do clinical trial. I'm short. Although it can molecule, we identify the molecule, then we have animal stage, then man clinical trial. And but other way, what we call reverse pharmacology is we know the plant is known by used by the travel people. It is in our traditional information knowledge. Even in Ayurveda, Unani, only we collect that plant and directly we can go for the validation of those extract. And if you find they are, yes, they are very prominent, they are giving good activity, positive activity, then we can go to directly to the our animal clinical trial. It means it takes hardly very fast, we can say it. If you compare it, Ayurveda Unani product, Ayush herbal product, we can make in 3 to 5 years. Only up to 20 lakh rupees worth of it is required for the doing trial, standardization, and animal study. And modern medicine, you can it takes 15 years or more, and you have to invest more than 10 crore. That is a modern medicine. That is allopathic. Although in between now we have a third group of drug in India after 2016, we call it phytopharmaceutical drug. It is in between these two, and a fraction which is active, but however it will take another 5 to 8 years, and fund is also required for that. So these two things are and now exist in India also for the development new drug. So better we can use this herbal medicine using Ayurveda, Siddha, Unani. We can call it, or we can call it herbal medicine. There are legality are there that also we can just example give you some example. Whatever you develop, you have to maintain quality, safety, efficacy. Three pillars are very important to demonstrate where my extract has a quality, what are the parameters we have to develop. Then safety is very important. If drug is safe in animal study, then you can go for efficacy. If it is efficacy but safety not there, it will not allowed by the clinician or by the drug controller. So quickly, I will go efficacy safety required for that. We go for pharmacological and safety study. In vitro, in vivo means in lab and in animal also we have to demonstrate, and limited safety we can do it in case of herbal medicine. And directly you can go to not clinical. If you want to have a study, field study, you can do it as per the ICMR guideline also. So quickly you have that drug. I will say you we have the etymologically plan identified, go for extraction, go for the animal study, standardize it, make a tablet, and quality control parameter. Then if you find active, then directly you can go patenting, or later on trial of that. and later on you can do marketing and whatever dosage form you want, you can do it. And globally you can export also as per the law of that country also. So this is traditional approach. Directly you can make like this also. It will take 3 to 4 years because we have done it also. So this is the for herbal medicine. Only thing the drug, whatever the plant you have, it has to comply with Drug Control Act 1940. We say if a plant is mentioned in Ayurveda, Unani Siddha, you directly go for its quality control and no efficacy required. Directly you couldn't go. Like moringa is mentioned in our text Ayurveda, you can make powder and directly you can make the powder packaging, although GLP lab is required, and you can go without any clinical trial, efficacy is not required for the market purpose. But if your claim is different, then you require this. So we take the best way, go for the plant which are mentioned Ayurveda, Siddha, Unani, and use this. This law and demonstrate efficacy and safety like this. This is a plant, you make the extract, only three extract allowed 50 hydroalcoholic, 50%, 90% water. These are allowed in Ayurveda, United States only, no other factor, only ethanol is allowed. Just you go for the in vivo study, in vitro study also, and do the safety. These are quality control parameter are required. You can do HPLC profiling, phytochemical, organoleptic correct. All these are required, and directly you can go for stability of the drug, and directly you can market it also, and after you can do patenting also. So this will require at least 2 to 3 years time. The plant should be mention all athletic books of the Ayurveda, Siddha, Unani, then only and for same purpose, same elements is required. So like some example as the Artemisia annua, Artemisia we identify. All plants, this is known in our traditional knowledge also. Poppy is also opium, morphine, codeine, all down injection are there. Texas, vinca. Like this, we have hundreds of plants are there. So this is very best approach to using our ethnobotanical leads directly. It shorten your time to save your fund also, money also, and you have more chance to get the best result. Like this is the artemisinin, this is Artemisia annua. The two use you got Nobel Prize in 2015, I think, in medicine because the plant is being used in China for the fever, malaria fever. And she identified this plant, isominin from this plant, and she got the Nobel Prize. So this is the reverse pharmacology, we can say it, and she proved it. Yes, it has the effect. It is a yes good for anti-malaria drug also. And what are rules are there? Safety, efficacy, clinical trial, phase one, phase two, whatever there, she had done it, and finally she got the award. Also. There are number of examples out here, but I will not go because time is short. Only quality is also important. You know, in the market you will get different type of material. If you maybe they have fungus also, other thing also. How to identify? And we don't know what type of quality is there, what type of active compound is there because of so many things. You don't have a right identification, right species. There is genetic variability also, growth variation, growth time also. Different ecological how ecology of what you call it, phytogeographical zone and different type of compound. So there is a very complex thing is there to how what the whatever raw material here to maintain that you have to have good chemist also who can do a good botanist also who can identify the plant also. So you require this. This is one example, just example I was in Saraka Asoka. This is the Asoka plant which we got Asoka Rist. Number of plants. But in market you can see polyalthia, whatever we have the revenue garden, bark of that is being used, Soria robusta being used, Mahonia. So this type of adaptation going on. Like Texas, Talis is this is the genuine official, but you may get the abuse. So there are number of example like Jatamansi. Two species are different, but they look similar and their activity different. So we should know what is the exact activity also. Even season, if you see this is a rhubarb, if you correct in winter, you will not get the activity because compound which their anthracene, they are only oxidized in the summer when temperature rises. These inactive anthrone convert into anthracene, which are active. So you have to collect a plant in the winter only. This type of information we need for the other hundred of plants are here. Best of time of collection is also important. Even diurnal variation, morning or evening, you can find different also. After 3:30, this digital purpurea have high content of glycoside because all the glycoside, glycon sugar part in night it convert, and later on when we have the photosynthesis there, then again it combine together. So this type of information we need. So I will quickly give you one example of this, like for phytogeographical variability. Different parts of the country we have more than 12 phytogeographical zone in India, from Kerala to Himalaya also. The desert is there. So one example I will give tribulus, gokru, which have protodiazine, tribulucin. These are compound acting compound here. Acurus calamus, yes, it has essential oil, beta essential. So we have collected all over the India. One in case of tribulus, more than 41 accession we have collected all over the India. It took 2 years, and we have done the chemistry of that. Now you can see variability. Maximum you will getting we are getting tribulucin, protodiazine from the desert Rajasthan area. You can see very little in Indo-Gangetic Plain, UP area. So best way to collect a plant having high content of tribulocin, go to the Rajasthan. Then this type of study we have done it because variability there we have to enter the best material. So this type of study is required. Similarly for Acorus calamus, yes, we have done it. Now you can see in short, in Himachal, we are getting 9.5% of oil. While in Lucknow, we have 2.3. So much variation. We have all over the India. So if chemistry is changing, alkaloid is changing, volatile is changing, naturally chemistry will, this activity will change. So this type of variability is there in the nature. So I am giving you only one example, two examples of this. This type of study will require in the future also. So where you get, suppose you have the plant, how to identify where the standard, yes, go to the monograph pharmacopoeia. We have reference book are there, collect right species from the nature and doing the drug evolution. Then you can either good raw material, quality material. To quality raw material, you have to have botanists who can do the macroscopic character, macroscopic study, and physiochemical parameter like hash value and moisture content. Later on, what type of compound it has. They can use by HPTLC, HPLC also, GC also. You can do DNA fingerprinting also to identify right species if there is a variation, and you can do the bioactivity. Outdoor active character are there by collect this. So this is the very basic things which are required to identify right raw material. So in the short, we need botanists, we need chemists also, and pharmacists for drug development. This is not a one area, and like botanist, he can identify all five species of basil. Different species are there, but under microscope you can see the different vein termination. But in short, these are the very good. Like Brahmi, we have two Brahmi, but one is like Bacopa, one is Centilla. By even the stem cell, the xylem cell is different. Their arrangement is different. So these are the very good character by which you can identify morphologically and anatomic also. This is Bhuyamla, we have Phalidamaru, Fraternal, and Madras Fraternity. But as a layman, they look similar. However, if you go under microscope, you can see different type of stomata, and there are the wavy subsidiary cells. They are all regular. So these are the characters we have developed. Margin also different margin by which you can identify right species. So these are the characters. And these are books which are, I mean, available. It is all government gazette under gazette also. This is legal document. We have more than 800 plant now, which have monograph, and we have contributed more than 67 plant for NBRI. My team has done it. So you can refer these books while identify the right raw material, quality raw material. All data is there, and all are legal one. Even I.S.G.B.R.D has done it similarly. More than 52 plant we have contributed. We have more than now 400 now. Different volumes are there where you get every detail of the plant, its chemistry, its activity also. This is good books for the future. I mean, if somebody want to work in this area, they have to have. This is the what Ansuma was telling. This is the BER 34 we have developed. We got technical award on this, and I think more than 300 crores worth of rupees has been. They have sold this in India also. That company. Similarly, we have made nowadays, I mean, in Manipur, we have made the ethnic hot and sour soup. That is not from the what we are getting in our nature. It has no Ajinomoto. All plant is from nature. Even it is not a citric acid. We identify some plant which are good antioxidant, having sourness also, and good for immunity, and make the soup also. Although base is that other raw material is there, and you can make the rice, brown rice powder also. So you can make that. These are the all five plants we have for vitamin C is there in this raspberry flora, Papa nigra, alien which is there. So these are the plants which are we have used for this making soup, and it has no synthetic compound is there. This technology I believe we have patented it. These are the all we have done the calorie value, sugar, vitamin A, vitamin C, all it is there. Similar, we have made the soft drink also for that. That is like Coca Cola, but not like it has all herbs are there. So this is technology we have. Like this, we have more than 32 patent on this. We have, and if some company interested, we can give you nutraceutical. Like this, we have toothpaste also, lip balm also. These are some example. Like this, we have some number of technology available for the slimming, for your wound healing, burning, and otherwise also. So we are working for the last 20 years on this area. And what I mean, short we can say, it's about the traditional and folklore information. Very important. If you go to get the fast way to get the good herbal medicine, and you can make combination of that by picking some information, automatic information. If plant is mentioned in Ayurveda, it is good to get the Ayurvedic license also on that. So we have to see what is the plant is there, information is there, you correct it, do the best combination, and directly you can do by doing the authentication, right species, and identify the botany character, chemical character is there, what type of chemical there is quantity. So installation important, and finally you can do the pharmacology, and finally the plant material or your product should be having no pesticide residue or heavy metal unmarkable load. If it is there, it should limit should be compliant as per WHO guide. So this is the way to get the herbal product, or whether it is nutraceutical or color or for the our medication also. This is the in a small short time I just touch a few points which are required. So a botanist is required, a pharmacist is required, a pharmacologist is required. These are the very important, and Ayurveda or Unani people are required. So it is a teamwork which give you the best results when we have so much diversity, plant diversity is there, and number of things are available in the people like in the morning session. I mean earlier, Dr. Agrawal also, Professor Agrawal also mentioned the name of Munda from Jharkhand. They have because he was praised like a god because he know the plant of that area also. They are he is cultivating, conservating, and giving it to people, travel people. So that type of people we have, and still they have. We have a number of people. That information now, all database is here. TKDL, I mean, we have the CSR TKDL library available. So we have that information. Now only thing right material, right quality, and whatever innovative product you want, you make it. And as per the our this conference say, innovation is there. Innovation means whatever demand is there, what are the gaps are there, you fulfill by using more scientific and your traditional knowledge. Thank you very much. I think in the short I will complete my lecture, although time is short, but some point I have mentioned. Thank you very much. Thank you.
Thank you very much, sir. It was great listening you, and I hope the students must have learned a lot from this. Though I really wished that sir could be joining us offline, but through this medium, I would like to share with you all that due to some personal reasons, due to some health issues of father of sir, your mic is on mute. See if you're speaking something.
Father is not well, so
Yeah,
I
so
could
we
not.
got to know
I,
that.
I, it is, I love to be there. It is good to have one to one. And the student, you can, I mean, the people know, the scientists know, but we have to, I mean, fulfill the appetite of the student, what to do, which direction they want to go. That is important.
Yes, I
And
completely
I,
understand.
with the
And
Professor Samuel also, I could not meet this opportunity I lost. Anyway, thank you, and Suman, and all the
I
delegates.
wish for faster recovery of Sir's
Thank you.
father, and I'll just request Professor Dr. Manjula Thomas, Madam, to speak something, and then I'll request Dr. Sakshi Walker, ma'am, to give the conclusory remarks, and we conclude today's program with this only. And I request everyone to join in tomorrow in the offline session, morning 8 a.m. at St. John's College, Agra. Over to Dr. Manjula.
And
Your mic is on mute, ma'am. Uh,
Thank you so much, sir, for very informative uh, whatever you have shared about the development of herbal drugs based on traditional knowledge, and very nicely in a very simple way you have delivered the history about how the herbal preparation has been prepared, about so many plants we have heard about Vasaka and Centinela, Texas, and so many other cosmeceuticals also. One question I have, sir, that artemisinin and quinine both are anti-malarial drug. So how artemisinin is more effective, or if you can please share about the comparison of this artemisinin and quinine. Is it better
yeah,
than
yeah, yeah.
quinine?
Yeah, it is why I am saying initially people are using quinine also, and slowly people are, I mean, not people means mosquito get the resistance on this drug also. Drug
Okay.
resistance is there, but because of so many decades of that we are using quinine. Now whatever the development is that they put quinine as a positive control also when they do the experiment. So when they see it is better than quinine and it is safe also, then only they claim it. Always think, I mean, development means whatever I mean, previously developed drug is there, make it positive, I mean, drug, positive control, and the compare with that. So Artemis is a new one which is not create any resistance. I mean,
Okay.
resistance is due to the quinine, but it is a
So,
novel one.
so sir, which part of the plant of artemisia
Whole
is used?
aerial part,
Roots?
all part. No,
All.
not
Okay.
root, aerial part.
Okay.
Aerial part.
Thank you, sir.
We
Thank you.
have in India we have cultivating. IPCA is the company. IPCA is the company which I mean supply this drug, and then our all in the north, south part Kerala, North Kerala also, now Sri Lanka also, Tamil Nadu people cultivate artemisia, and it developed by the variety developed by the CMAP also, the CSR labs.
Thank you, sir. Thank you. And I'm sure that all our participants have benefited through your very informative talk. Thank you. Over to Sakshi, Dr. Sakshi.
Yes, ma'am. Thank you. Hope I'm audible.
Yeah, yeah.
Yes.
Yes. Um, a big thank you to all the speakers who joined us today, this afternoon of deliberations, of bested talk, of um, all brainstorming that we could do on sustainability, on how we could reach sustainability, of what is required right now, what is the need of the hour. So, thank you, all the speakers. On behalf of ISGBRD and St. John's College, the organizing committee, I thank all the speakers. Um, as I can see, I mean, there are so many comments that I can see on the side panel of my laptop. Students, they want to ask so many, they have so many queries. So I guess it all tells us that the sessions that we witnessed, they had discussions on frontiers of applied sciences, insights on sustainability, how judicious use of natural resources, water management, eco-friendly technologies are essential to ensure food and livelihood security for future generations. And of course, this enlightening discussion on use of local traditional plants followed all these sessions, which threw open avenues of research possible. The list goes on and on. I think my paper will end if I write down all the points. So I think Anshumanji, the session has been today's conference has been a resounding success, and we have all been able to absorb and realize where we are at fault and what is the need of the hour. So, thank you once again, all the speakers, and of course, ISGBRD for bringing forth this the need of the hour, uh, what we need to do right now, and how we need to achieve sustainability. Thank you so much.
Thank you. Thank you. Thank you, sir. And I will take leave also. Good night.
Thank you very much, all. And with this, we conclude the program.
Thank you so much, each and everyone. Bye bye. Good night.